Okay, well, I think we're good to start. So welcome everyone to the CTX 2024 Scientific and Family Meeting. And the meeting is, of course, hosted by the United Leukodystrophy Foundation and organised by the CTX Alliance. And I'm acting as the meeting chairperson, along with Jean Pickford, who helped organise as the executive director of the CTX Alliance. And it doesn't seem like a year since all of us were last here at the 2023 CTX Scientific and Family session. And I'm glad to see many of the same faces and welcome new faces here. It was an excellent meeting last year, and I'm excited about the agenda this year. I'd just briefly like to mention some of the highlights achieved by the CTX Alliance over the last year. So we're very proud that we organised and hosted the International Scientific Meeting in August 2023 in Jerusalem. And the overarching goal of this meeting was to catalyse essential international basic research and clinical collaborations for CTX, aiming to advance the understanding and enhance outcomes for CTX patients and their families and foster patient group partnerships. And it was a very successful meeting. Lectures and discussions from esteemed researchers, medical professionals and leaders of patient advocacy groups participated from 11 countries. There were 109 participants, including 75 in person, 16 attending virtually, and 18 local CTX patients from Israel, constituting the largest international gathering of CTX experts to date. And we had the privilege of presenting a Lifetime Achievement Award to Dr. Vladimir Berginer, recognising his groundbreaking contributions to CTX research and his commitment to improving the lives of CTX patients and their families. And so this is just a photo here of the gathering. And again, there's a number of people that we have here again today. Another highlight is that Mirum Pharmaceuticals acquired chenodeoxycholic acid from Travere. And most of you know this. From the perspective of the CTX Alliance, the transition has been smooth. There's been no change in customer service and access to chenodeoxycholic acid. To this point, the financial and patient advocacy support continues at the same level as it did with Travere, which is great. And we gratefully recognise their generous sponsorship of today's meeting. Something else that the CTX Alliance has been working on is the newborn screening application for nomination of CTX for consideration for addition to the recommended uniform screening panel of disorders that screen for in newborns. And so we have a newborn screening task force of leaders and experts collaborating on the nomination package. And we just had a meeting this morning. We're hopeful that we should be able to submit at minimum the first part of the nomination by the end of this year. So that's very exciting. The new nomination that just became available here in May consists of answers to a 4-question preliminary nomination form, which, as I said, we'll be hoping to submit by the end of the year. And then a full application package if the Secretary's Advisory Committee for Heritable Disorders in Newborns and Children approves the first preliminary nomination form. So after submission, patient involvement will be critical to reinforcing the importance of newborn screening. And I know Bobby Blanchard was there at the meeting this morning, and Jean have been working with other patients and families who are interested in helping support this. So if you're interested, please do reach out and talk to Bobby about that. I'm going to give away. So there'll be more information to come on this. Another thing is the neuropsych survey. The patients and families have said that this is something that's very important to them. So one of the focuses over the last year of the CTX Alliance has been setting up a survey to kind of assess this and be able to make this information available. So currently we have a live English version, a Spanish, Hebrew and Dutch versions are to come. Is that correct, Jean? And the purpose is to gain a better understanding of the neurologic and psychiatric burden of disease when living with CTX. because to this point, very little information has been published about the neuropsychiatric burden, yet increasing conversations about this associated symptom. We've had many of these over the last years at the meetings. This data will be reported in industry publications and other relevant areas to bring attention to and stimulate research for improved patient outcomes in this area. So this is today's agenda. And I'm very excited and grateful to have so many great speakers here. So our first speaker will be Dr. Florian Eichler, who comes here from Massachusetts General Hospital and Harvard Medical School. And he'll be talking about the importance of clinical trials for drug safety, efficacy, and approval as we think about potential future clinical trials for CTX. We welcome Dr. Gloria Gonzalez, who a colleague of hers spoke about their project at the Jerusalem meeting, working on potential gene therapies for CTX, and presented data from the mouse knockout model for CYP27A1. And she'll be attending and presenting remotely. And then we have Dr. Irina Pikoleva from Case Western Reserve University, and she'll be talking about inhibitors of CYP27A1 based on the enzyme assays that she has set up in her lab. I'm also excited to have Dr. Lei Ling here from NGM Biopharmaceuticals, and she'll be talking about a variant of FGF19, that's aldafermin is the name, to treat cholestatic liver diseases and its potential utility for bile acid synthesis disorders. Then we'll have a break, and the time of the agenda is being changed slightly. Then we'll have Dr. Sipora Felix-Zakai from Bar-Ilan University in Israel talking about real-world data supporting the use of chenodeoxycholic acid treatment in pregnancy in CTX, because there's been some question about this. Then we have Dr. Kenneth Setchell from Cincinnati Children's here, who will be talking about his experience using cholic acid to treat CTX, and then potentially using co-administration of cholic acid and chenodeoxycholic acid. And then we have Dr. Austin Larson from the University of Colorado School of Medicine, Aurora, Colorado, who will be providing an update on the recent RESTORE clinical trial supporting the use of chenodeoxycholic acid to treat CTX. And then finally, we'll have Dr. Zhentao Zhang, I'm not sure if I pronounced that correctly, who'll be the last speaker participating remotely, hopefully. It's very early there in China at the moment, and he'll be talking about some of the ways that cholestanol may cause its pathology as part of CTX. So welcome, everyone. And with that, we'll start with Dr. Eichler.
Excellent. Thanks, Andrea, and delighted to be here. You've built such a wonderful community that's international, as we see from people attending here. It's also the Jerusalem meeting was outstanding, so I was really happy to see that. I think Gavin is pulling up my slides. While slides are being pulled up, the task for me is to be brief, be sweet, be gone, as my mother used to say. I'm going to highlight the landscape of clinical trials and why we conduct clinical trials. I'm a neurologist at Mass General, so my expertise is in neurological outcome, thinking about neurogenetics, but I come here every year to learn biochemistry from Andrea. So I'm not an expert in bile acid synthesis, but I've learned from many of you over the years. So I will, without further ado, go into some of the thinking. Oops, just tell me if I'm pointing. Okay, disclosures briefly. Okay, why are clinical trials important? So hopefully the slides didn't get cut off. In short, we distinguish between observational trials and interventional trials. Trials in general are here to evaluate new medical interventions, including new drugs, devices, vaccines, lifestyle modifications. In short, it tests something that has not yet scientifically been proven or is known. And I think this sort of equipoise between, of not knowing is very important as we think about new interventions and how to appropriately frame things and think about risk benefit for patients. And one of the big questions we have is how do we know whether something is safe and really works? And the main goal of clinical studies and trials is to avoid bias to arrive at trustworthy answers for families. It's as we all attempt to embark with best intentions for our patients, there's a good chance that we might introduce bias with our good intentions. A good example might be that unbeknownst to the physicians, they're enrolling patients who are healthier into a new treatment group and leaving the patients who are more severely affected on placebo or usual treatment group. And that would then create the impression that a new treatment is more effective than the usual treatment. But that is, of course, here simply a mistake of the enrollment, and hence the need to randomize trials and to control trials. Randomization helps reduce bias and eliminate differences between treatment groups by randomly assigning patients to either treatment or placebo, or treatment versus standard of care. And another tool we have here is stratification, which means that when we're enrolling in these different groups, we try to do this in a balanced way so that not, all the severely affected patients are in one group versus the healthier patients in the other. And this stratification is very important and helps in even the smaller trials I've run. I've always done block stratification, block randomization in order to ensure that these arms are balanced. What are the phases of clinical trials? We know about a lot of the preclinical studies that are often needed, the proof of concepts that are provided, toxicity studies that go into first design of trials and in phase one. Usually phase one looks at safety and gets some information about pharmacokinetics, like how a drug interacts with the human body. Phase two is then safety and dosing. You're looking at also some elements of efficacy. Does this drug actually work? Is there a signal here that is supporting of that? And then phase three is usually confirming whether the drug really works and to provide further safety monitoring. All of these different phases are necessary to then get approval by FDA. And there's then even after approval, post-marketing, safety and efficacy. Often the numbers used in these different phases are quite large in conventional trials. It's difficult to detect significant differences with small numbers in rare diseases. So that is a challenge I'm going to touch upon briefly here. How do you optimize clinical trials for rare diseases? So the challenges here obviously are smaller patient populations. Within that population, there might be clinical heterogeneity, yet there's huge unmet need, and you cannot really wait to find larger numbers of patients. You have to work with the population at hand. And there are different ways to minimize the size while maintaining statistical significance. We often think about adaptive trial designs, crossover designs, where we allow patients to be enrolled in a trial where you have treatment versus placebo, and then you cross over so that every patient during this trial does see treatment, although also placebo. One advantage you do have in rare diseases is that there are more robust biomarkers and outcomes than in more common diseases. So you have this disease-causing genes having a direct impact upon biochemistry can be a huge advantage, and also allow for you to argue that this is representing sort of a real change in the pathology. Precision medicine in rare diseases also allows for gene therapy trials, and that becomes a bit of a different animal, so to speak. Usually gene therapy trials combine phase 1, 2, and 3. So you're always looking at safety and efficacy as you're going for approval. You're continuing to enroll. Often placebo is hard to achieve, particularly with progressive diseases, and gene therapy is often a one and done, which can sound like an advantage. But on the other hand, it is something that once you give and once you administer, it's hard to take away. And so how to think of that when you have a rare disease and you have a potentially vulnerable population and you're dealing with new experimental treatments. Important part in designing clinical trials is how to define outcomes. And the first thing you want to think of as you are designing a clinical trial is, will this intervention, will this treatment change the patient's life? And FDA has a very concrete North Star for that. They say it should make the patient either feel better, function better, or survive longer. And that's their simple definition. You have to hit one of those three or several of them to convince FDA that this is really something that should be approved. Not all trials are FDA regulated, but the FDA has a helpful framework. What that means is that if you are proposing trials with already FDA-approved drugs, you're looking at something called repurposing of a drug, that it's already been used for a different indication. Sometimes you have to run it past FDA, but you don't necessarily have to submit an IND in all cases, which is an investigational new drug submission. I just want to highlight here that, again, the theme of biomarkers, because I do think for rare disease and for CTX in particular, this is so important. Biomarkers directly measure something related to the disease, only indirectly related to clinical progression. So while it is an advantage that it is directly often related to the disease-causing gene, we have to be cautious that it might not directly correlate with progression. That's always a tension that we're thinking about. So importance of biomarkers. When we speak about biomarkers, it's really not just one type of biomarker. I stole this here from Andrea's poster yesterday, as your mentees taught me again about 7-alpha, 12-alpha, C4. I'm always trying to repeat the biomarkers so I can finally come back and sound more learned about this. But the beauty of this is how nicely it really separates some of the carriers here from the CTX patients. And that is, of course, so key when you want to have a solid diagnostic biomarker, and everything that is being undertaken here in the newborn screening world is really key to giving confidence in this disease entity and ability to monitor. But you have to keep in mind that just because it's a diagnostic marker doesn't mean that it is necessarily prognostic or predictive of future outcome. And I think this is where we have to be careful and think, can we really think of it as a surrogate, something that would stand in for the ultimate pathology? Maybe there are imaging biomarkers that we need. Now in neurology, there's a lot of talk around neurofilament light chain. Is that something we should be looking at longitudinally to understand neurological disease progression and a biomarker thereof? So just some thoughts here. clinical trial oversight, just to highlight, when we start trials, we have institutional review boards, we have data and safety monitoring boards, and sometimes patients come to me and say, why all this administrative burden? What is this really for? We really want to ensure that risk subjects are minimized. It's very important that patients understand what they're committing to as they step into a trial. Informed consent is a way of you listening to this and signing documents that really show that, that this is in your interest and happening with your consent. Data safety monitoring boards have become a big part of my life, as I'm always in gene therapy trials, waiting for the review of the DSMBs. They are really safeguarding interests of the study subjects, making sure that as we go to the next dose, as we go to the next phase of the trial, everything is safe and that the integrity of the trial is intact. So while it may seem cumbersome, it is really important for patient safety, for patient well-being moving forward in these experimental trials. So not to carry Alice to Athens, but you all know of this already huge, important insights that I was actually very fortunate to see Vladimir Bergner in Jerusalem again. Had originally met him here at ULF decades ago, and to just reflect upon that accomplishment to bring about chenodeoxycholic acid and the ability to lower cholestanol is huge. The question, I guess, for me today is, are there other treatment approaches to consider beyond this? We know this is an approach approved in Europe, but here still not yet by FDA. But is it address all aspects of CTX, and how do you think about the treatment approaches overall in neurometabolic diseases? And I'm just going to frame this by saying, in general, I think about disease-modifying therapies, everything from enzyme replacement to cell transplantation, substrate reduction, gene therapy. We are getting more and more into this era of expanded access therapy. It's a very difficult realm where we have to carefully, patient by patient, decide on whether it makes sense. And then I think very important area is symptomatic therapy and understanding prevention versus management. Just briefly here to highlight that in all neurometabolic diseases, there's a DNA, a gene defect, RNA defect, reduced protein or enzyme function. There's either lipid buildup or metabolite loss. You have these then causing cellular mechanisms from oxidative stress to inflammation to impaired mitochondrial function, neuronal loss, and tissue damage across many of the leukodystrophies, ranging from affecting myelin to axons. And if you look at this diagram, you can think of all of these steps having a corresponding intervention, whether it's gene therapy affecting the DNA level, antisense oligos, RNA therapeutics at the RNA level, enzyme replacement, substrate reduction, or small molecules working further downstream. And I'll just give you some quick examples in the field of lysosomal disorders, enzyme replacement. First success for Gaucher, applied to Pompe, Fabry, and MPS. Tay-Sachs and Sandhoff disease, we've seen this not work, and really penetrating blood-brain barrier has been a huge challenge, even for metachromatic leukodystrophy that tried to administer this intrathecally. This has been difficult. Substrate reduction therapy we've seen in GM2 gangliosidosis, miglustat, venglustat more recently, also not fully panning out, unfortunately. So while reducing the burden of accumulated substrate, it has challenges, and all the more reason to celebrate what Chenodal has accomplished in terms of this feedback inhibition that can happen of a cholestanol. What approaches to gene therapy exist? So you'll hear after this read some exciting new work on gene therapy that's liver-directed. I'll just say here, we distinguish between in vivo gene therapy that is usually mediated by adeno-associated virus, which is a direct delivery to target organ, whether it's the liver or parts of the nervous system, and ex vivo gene therapy, which usually depends on you harvesting cells, and often those are bone marrow cells, and then they're transfected with a lentiviral vector, delivering a healthy copy of a gene and then delivering them back. So these approaches may not make sense for cerebrotendinous xanthomatosis, but I just want to point out this landscape and this evolving progress in the field, because I do think FDA is becoming more permissive to having some of these new approaches come to patients. So again, briefly, adeno-associated virus, and you'll hear more about this later, which it allows for direct delivery using this smaller DNA virus, and think of this as a delivery system that then carries a healthy copy of the normally defective gene to the patient, and thereby impacting the disease process. And different here with the lentivirus that usually requires using it outside of the body upon cells that are harvested from the patient and then returned back. So here's some examples of the kinds of trials using adeno-associated virus we've had in our hospital recently in Canavan disease, using an intravenous approach to deliver genes to patients. Now that means, of course, going into the bloodstream, and going into the bloodstream means that you can also see immune responses, and this is something that can be dangerous, and we have to be careful about that. Another reason to have clinical trials, so there is oversight, so there is monitoring, so that we don't put patients at risk. And just another example of a gene therapy that was approved for another leukodystrophy using gene therapy of bone marrow cells, so taking advantage of the crosstalk that exists when cells move from bone marrow to blood and from blood to brain. Just also want to highlight that on the level of the RNA, we have now some new modalities that are called antisense oligonucleotides. These are small chemically modified single-stranded DNA that have been used. You might have heard this in spinal muscular atrophy, where this has been quite transformative for patients when it's administered into the spinal fluid space. And we are seeing now a whole array of individual attempts at correcting rare diseases. So a lot of safety aspects need to be considered for this. There's inflammation that can occur due to the AAVs. We have to be careful about the intrathecal ASO administration in terms of complications from increased intracranial pressure and other aspects. So just a few words on a huge wave of new technology that's heading our way with CRISPR gene editing. We're going to see this probably in every field of genetics happening, where we're going to ask ourselves, can we really precisely correct the gene defect in individuals, and how to scale this up to patient populations in need is a huge question for FDA and the NIH right now. And of course, we want to do this safely so that, again, there are no off-target effects and we don't see side effects. Okay, so I'm just going to close by just saying some of these expanded access opportunities with single patients are becoming possible, but often they just provide access and they don't give you evidence of whether this is really working. And so I would remain a strong advocate for clinical trials and for understanding and focusing upon what matters to patients, which is largely related to symptomatic therapy. As highlighted before in the literature, we see very little reported on psychiatric symptoms, and this is a big gap, and I'm happy to hear that now in the surveys on neuropsychology. We'll be closing this. We compare this with our MGH cohort, where we see that a lot of symptoms are not reported, particularly around psychosis, dementia, learning difficulties, agitation. So can we learn from clinical practice? Can we make use of the multidisciplinary approach? And are there open questions that may prompt clinical trials? Same goes for management of these symptoms, also across physiatry, use of muscle relaxants, things that are like occupational therapy, psychiatric programs to help with the daily problems around behavior and disinhibition. We have still very little guidance. So just to conclude, clinical trials are necessary to assess what is safe and what works and to help prevent bias. New genetic medicines allow precise targeting of genes, but carry risks and need to be applied in specific contexts. What I want to say with that is you're not going to treat everybody with gene therapy. You're going to treat a very specific patient who, where it is really warranted that risk-benefit allows for this approach. And I think one has to carefully think about how we would select those patients. And if it turns out to be safe, maybe in next steps, things could open up and this could become broader. I think in the meantime, it's very important to think about symptomatic treatment at large, what is needed at different disease stages, not just for those in the early stages where we think we can make the most difference, but how to really deal with the unmet need in the advanced stages as well and give guidance so that families feel supported in the care for their loved ones. So, Andrea, I hope that was what you expected, but okay.
Thanks very much for a really wonderful introduction to the talks today. Are there any questions? And a really nice, you know, as you say, in CTX, we have a standard of care, which is treatment with chenodeoxycholic acid, but there are a subset of patients identified later that still continue to deteriorate on CDCA, so that we, you know, are interested in exploring other treatment options. So with that, I will open the presentation for Dr. Gonzalez. Do you have that available?
Sorry, I'm going to share my presentation. That's possible. So first of all, I would like to thank you for the invitation, for giving us the opportunity to present the work we are doing on gene therapy for CTX. And thanks to Florian for the wonderful introduction to gene therapy, because I think it's a good starting point. So, well, I'm my disclosures. I'm Biotherapeutics co-founder and CSO, and I'm going to try to guide you through what is gene therapy first, just very briefly, and what are adeno-associated vectors and how we produce adeno-associated vectors. Then the adeno-associated vectors that we use to treat CTX, and some experimental results that we have recently obtained optimizing the vector that Ruben Hernandez Alcoceba presented last year in Jerusalem, and to finish with conclusions. So I think just as a very, very, very simple and brief, our DNA is very similar to a computer program, and our cell is our computer. So usually the DNA, which is in our chromosomes inside our cells, has the information that is going to indicate and to guide how we are. And usually this information, what it does, and the variability on this information, what it does is to make us different. So everybody of this in the room and everybody in the world is different because the DNA is slightly different. So this is the beauty of the DNA, that this introduce so much variability. But sometimes the DNA has some errors. So the proteins that needs to be produced are not properly produced, and mutations and variation in the DNA can cause diseases, such as the case of CTX. So what is gene therapy, or when we start thinking about gene therapy? We start thinking about gene therapy around 1960, 1970, when we discovered what was DNA, how was this molecule, and more importantly, how we can be able to modulate this molecule. And we found different tools that allow us to cut and paste DNA. So we can cut and paste the information we have inside our cells and create new information. And for me, this DNA and playing with the DNA, making these new molecules, remind me the way that the old fashion of making films, no? So you have long films. I mean, you have a lot of images that you go to the edition and you cut and paste to create new images, new information. And this is exactly what we can do with DNA, because it's a very stable molecule. So we can take pieces of DNA and put all of them together to create this new information. So this is how we create functional molecules that will produce functional molecules using just standard molecular biology techniques. But once you have this molecule, there is a very important part of the gene therapy, that is the introduction of this material inside the cells. And the cells are very well protected, because since it contains everything we need for life, they have several membranes that are protecting the content of the cell, including our DNA, and it's difficult to enter inside the cells and enter inside the nucleus of the cell. And for this, we need vehicles. We need a way of transferring this DNA inside the nucleus of the cell, a way to introduce this information inside the cell. And the best vectors, or one of the best vectors or vehicles to introduce this genetic material into the cells are the viruses. And viruses, if we think about what a virus does in nature, it's just to introduce their genetic material inside the cell for their own benefit. No, they introduce the genetic material to produce their own proteins and to produce more viruses. And this is what they do. They don't do anything else but producing their own proteins and produce more genetic material. So the same way we are able to create these new molecules, these new DNA molecules, we can also manipulate the DNA that is inside the viruses. And in this way, we can create recombinant virus to treat diseases. And they are different type of vehicles that we can use, like adenovirus, lentivirus, adeno-associated virus. And also we have some vehicles that are not non-viral, but they mimic what a virus does, no? Like they try to mimic the same capacity that the virus have to enter into the cells. And as already has been mentioned, this can be done ex vivo. So we can take the cells from the patient, transform these cells with these recombinant viruses and introduce back into the patient, or we can just administer directly these recombinant vectors into the patients. And one of the most often used recombinant virus in the clinic, with right now 7 different products approved for the treatment of different diseases are adeno-associated viruses. And these viruses has some characteristics that explain why these recombinant vectors has been able to reach for some indications the market. And these viruses, as you can see in the figure, they are very, very small viruses that they have this icosahedric structure, and they were discovered as contaminant of adenoviruses. So the big viruses here are adenoviruses, and the small one is the recombinant adeno-associated virus. For this reason, was called adeno-associated, because it was discovered in the production of adenovirus. And which are the characteristics of recombinant adenovirus? As I said, they are very, very small. They have a 20-25 nanometers, so they are really small. They are one of the smallest virus in nature. They have a genome that is a DNA genome of a single strand, and they have one particularity that makes this virus very attractive for gene therapy, and is that it's naturally replication defective. So the AAV alone cannot replicate. The cell needs to be co-infected by another virus, more complex, to replicate. So once we inject one adeno-associated virus, this virus will stay only in the person that it has infected. It cannot replicate and pass from one person to the other. And another, and maybe the most important characteristics of adeno-associated virus is that they are not associated with any pathology. So you can be infected by a wild type adeno-associated virus, but you don't know because it doesn't cause you any disease. So they are non-pathogenic naturally. On top of that, the inflammatory response they cause is low, the immunogenicity in terms of T-cell response is low, and they are very easily manipulated. So the genome can be easily manipulated to create recombinant virus. And this is a schematic representation of the genome of a adeno-associated virus. It has only two genes, the gene rep and the gene cap, one that encodes the proteins involved in the replication of the genome, and the other in the formation of this icosahedric capsid, okay? And in the end, it has a hairpin structures, very small hairpin structures that indicates that this DNA is a viral genome. And these are the only structures, these ITRs, inverted terminal repeats, are the only structures that are needed to produce a recombinant AAV vector. So we remove the viral genes, the rep and the cap gene, and we substitute this material with the therapeutic material, which is usually the therapeutic gene, the gene that is mutated in the patient, and some regulatory sequences that will allow the expression of the protein in the cells that needs to express this therapeutic protein, okay? So this is the process to produce a recombinant AAV vector. We remove the viral genes and we substitute those genes with the genetic material we want. In this case, the gene to treat CTX, which is the CYP27A1. And this is a work that was performed in our laboratory, in which we treat CTX animal models with a recombinant AAV vector that has the ITRs, as I already mentioned, the regulatory sequence, which is a promoter that allows the expression of the CYP27A1 protein only in the liver, and the polyadenylation sequence that is also a sequence that is needed for the production, the correct production and expression of the gene. With this vector, we treated CTX mice, and we were able to reduce cholestanol and 7-alpha-C4 levels, as you can see in these graphs. So in black are animals untreated, and in the different colors, in the red and in the pink color, we have animals treated with the vector, and in gray, healthy animals. So as you can see here, and it's more clear, with 7-alpha-C4, CTX animals in black has high levels of 7-alpha-C4, and the animals that were treated has high levels of 7-alpha-C4 at the beginning, previous to the treatment, but after treatment, the levels went back to wild-type levels, to healthy levels. From these results, we decide to go ahead, and with having in mind the possibility of bringing this recombinant AAV to the clinic, we decide to optimize this vector to make a vector that is expressed more protein, that is less immunogenic, because there are some sequences in this vector that can activate the immune response in the host. And for this, we decide to modify a little bit the genome and make, as I said, a vector more efficient. So this will allow to improve the efficacy, the safety, while at the same time reducing the dose of vector we will need to administer in the patients. So for the reason, we create a new version of the gene that we call a codon optimized number 1. And as you can see, this version of the therapeutic gene express more than the wild-type protein, and we introduce this into different AAV vectors. And we test these vectors and we compare this vector with the previous vector, in which we have already observed a therapeutic effect, but we were reducing the dose in order to see if we can really improve the gene therapy for CTX. And these are the results. The first result we obtained analyzing the concentration of 7-alpha-C4 in plasma. And as you can see here, while the animals untreated has very high levels of 7-alpha-C4 in all the groups, we were able to reduce these levels, particularly to normal levels in male mice, the 2 weeks and 4 weeks after treatment. And we also have a therapeutic effect in females, although this reduction was not as significant or as big as in males. But from this study, we decided to go ahead with the vector expressing CYP27 alpha A1 codon optimized 1, because we didn't see really big differences with the previous vector, but this vector was more safe because it has less immunogenicity. So with this vector, we decide to go ahead. This vector is called CTX806 and is the product that we are now developing to bring to the clinic, hopefully to the clinic. And for that, we need to perform a series of studies, like identification of the therapeutic dose, long-term efficacy, as well as safety in relevant animal models. And I'm going to show you here some of the results that we have obtained very recently, and that makes us very optimistic in the sense that we are really observing a very important modification into the mouse phenotype. So here you can see that, as I already show you, we are able to reduce the 7-alpha-C4 plasma levels in male and in female mice to wild type levels. Okay, so this is a dose response study in males with a dose as low as 2.2 10 to the 12. We are able to normalize the levels of 7-alpha-C4. In females, we need to use a little bit higher dose, but at 4 weeks after treatment, we have in most of the animals undetectable or nearly undetectable levels of 7-alpha-C4. What was more important is that we also observe the complete disappearance of 7-alpha-C4 from the liver of these animals at 4 weeks after treatment, both in males and the females. So you can see that at 3 points 10 to the 3.3, 10 to the 12, 5, 10 to the 12, we are able to really eliminate completely this metabolite from the liver. And more importantly, the same is true in the brain of males and female animals. As you can see, even at the lowest dose of the vector we have used in this study, the 7-alpha-C4 is eliminated from the brain, while it's clearly detected in animals untreated, in the cow CTX animals treated with saline. And one thing that we were very happy also about is that, and this is very preliminary because we didn't know if we will be able to detect 7-alpha-C4 in tendons, in which usually patients develop xanthomas. And as you can see here, this is just 2 animals per group, in the group treated with a higher dose and only females, but we can really detect the accumulation of 7-alpha-C4 in the tendon of these animals that disappear when the animals has been treated with the therapeutic vector. So the results looks quite promising, and in fact our conclusion are that BTX-806 reduce 7-alpha-C4 in plasma in a dose-dependent manner. In normal levels can be achieved at 2.2 10 to the 12 VGs per kilogram in males and 5 10 to the 12 VGs per kilogram in females. BTX-806 can eliminate 7-alpha-C4 from the liver and from the brain in males and in females at all the dose tested, and our preliminary studies indicate that BTX-806 eliminates 7-alpha-C4 from tendons. So as you can see, this is a study that is still ongoing. We need to analyze more metabolites, but so far the results looks really promising. Next, we are doing, as I already mentioned, long-term efficacy studies and also combination studies with CDCA to see if we can combine both treatment with the safety conditions, and then remove the CDCA and maintain the long-term efficacy associated with the administration of BTX-806. So, and this is my last slide. This work has been, the main responsible for this work has been Laia Trigueros Motos, Blanche Shantamarita, and Celine Bouquet from Vivet Therapeutics, Athima, Angie Molina, and Ruben Hernandez Alcoceba, together with a bunch of people that are taking care of analyzing many things, many aspects that I didn't mention in this presentation, but we are also analyzing neurological involvement in those animals and if we can correct it. And for us, it was really key the analysis of all metabolites that are performed at the Sorbonne University by the team led by Antonin Lamassière. And of course, this is not possible without the funding that we have obtained from BPI France that that is supporting this study. And this is all, and I will be very happy to answer your questions.
Oh, thanks very much, Dr. Gonzalez.
Thank you.
Thank you for sharing that very nice data with us. Do we have any questions, Irina? Here you go.
I'm wondering whether you tested what organs your gene therapy increased CYP46A1 expression.
C27A1.
Yeah, you do gene therapy. You expected to increase CYP27A1 expression. Did you identify the organs which have increased expression?
Only the liver, only in hepatocytes, because the construct has a liver-specific promoter. So it's only expressed in hepatocytes. The vector can go to other organs, that's true. It's true that also that this is a very liver-tropic vector, but you always have some distribution. But due to the inclusion of this promoter, we have only expression in hepatocytes.
Yeah, Ken Satchel from Cincinnati. That's really impressive work, particularly on the C4 levels, which of course are a surrogate marker for bile acid synthesis. But can I ask you, did you also look at what changes occurred in the composition of the bile acid pool? This
is a study ongoing. In fact, in the dose response study, we will do a full metabolite analysis to see how if we are able to normalize all the metabolites. In fact, in a previous study, we compared the effect of CDCA with the gene therapy vector, and we see very interesting differences in the way that with the gene therapy, you recover the homeostasis, the normal bile acid metabolism. But yeah, these results will be ready end of the year, more or less. But yeah, we are really looking for that. Yeah.
A great talk, Florian Eichler here. So I was curious about the differences between the males and the female mice. Do you have an explanation for why you're seeing this? And also, at what ages are you treating mice? I'd be just curious.
Yeah, we are treating young animals. These are 8-week-old animals. Okay. It's true that in CTX, the liver is healthy, even in older animals. So we think that this therapeutic effect will be maintained with time. And sorry, your question was
on gender.
Oh, gender, yes. This is something that is not particular to this the CTX animal model. It's particular to mice. There is AAV vectors has a gender different in the levels of transduction and seems to be related to hormones. And in fact, there is a very old paper. Now it's like maybe more than 10 years ago, in which they have performed studies in in which they have removed the ovaries, and they eliminate these differences or reduce these differences. But it's something that has been reported in mice, but not, for example, in non-human primates. So we need more data from clinical trials to see if this is something that happens or not in patients. But
so you say the protein expression is the same that you're delivering, or
no. There is also a difference. Yeah, okay. Transduction is different. Protein expression and mRNA levels are a little bit lower, always in females.
Dr. Gonzalez, this is Jean Pickford. There's 2 questions in the chat. The first one is, thanks, very exciting news. When do you plan to start studies in humans? And when can this therapy be approved?
Yeah, I mean, our goal is to generate all the data necessary to fill IND 2026. So yeah, we still have 2 years, 3 years of work to go through all the safety, production, etc. But yes, next year we need to be ready with all the preclinical data, all the preclinical package. Yes.
Okay, we have one more question. Do we know if the affected bile acid synthesis pathway in CTX is only in the liver, or is it in other organs, example, the central nervous system, GI tract as well? Wondering if treating the liver would address the symptomatic symptoms of CTX.
Yeah, that's a great question. I think this question, I mean, the CTX is really the expression of CYP27A1 is mainly expressed in the liver. So we think this is the main organ and the reason for the accumulation of the metabolites. But it's true that probably there are more people in the audience that can answer better than me.
I was just going to say, so Dr. Piccoliva, who's talking next, I'm sure she has a slide that looks of expression of the enzyme all over the
Great
body.
segue.
And we just have one really final quick question.
I don't know, maybe, hi, great talk, thank you. Maybe I missed it. Are you planning to give one-time injection or is it like several times? How often?
Yeah, I mean, recombinant AAV vectors it's true that they are non-integrative vectors, but they remain episomal for very long time. If you look at the data on using AAV for the treatment of hemophilia B or A that are already in the market, for hemophilia A, they have shown that you can express factor 9 to stable levels for more than 7 years. So more than 7 years because this is the longest follow-up. We don't know for how long they will maintain the expression. So this is our goal, to reach at least 7 years of expression of the therapeutic protein. But if this is true, and if the expression of the protein decay, we hope to be ready to re-administer a second dose of the vector at the time that this therapeutic effect disappear. But this one shot.
Okay, well, thank you very much again. We'll
thanks to you.
Thanks for staying up so late, and hopefully you'll stick around for the rest of the
Spain,
meeting.
This is not very late. Don't worry.
Oh, it's not okay. So our next speaker will be Dr. Piccoliva. Okay, and that was a very nice segue into her talk, and she'll tell us all about where CYP27A1 is in the body.
So should I use this microphone? Right. Wait, first of all, let me see. Okay, so first of all, thank you for the invitation and opportunity to share some of our data with you. And today, at first, I will be talking about CYP27A1. Let me see. Okay, CYP27A1.
Can you talk into your microphone?
CYP27A1. Then I will. Can you hear me?
That's better.
Okay. Okay, then I will explain the link of CYP46A1, another cholesterol metabolizing P450, and CYP27A1. And then I will present some of our data on CYP46A1. So don't let me. Okay, and let me start by giving a very brief background biochemical information on CYP27A1. So CYP27A1 is a ubiquitous enzyme, meaning that it is found in almost every cell in our body. However, it's expressed at very different levels in different organs. And more importantly, in different organs, CYP27A1 has different substrate preferences. So in many extrahepatic organs, including lung, including brain, including vascular endothelium or our blood vessels, including macrophages, CYP27A1 metabolizes cholesterol and introduces hydroxyl group at the C27 position. And therefore, CYP27A1 is also called sterol 27 hydroxylase. However, in the liver, we all know that CYP27A1 metabolizes bile acid intermediates. And then in the kidney, CYP27A1 participates in the activation of vitamin D3. So, and we all know that loss of function mutations lead to CTX. But perhaps what is less known about CYP27A1 is the link between CYP27A1, 27-hydroxycholesterol, and hormonal breast cancers. Hold on.
I thought it takes the alcohol to the acid also, or no?
Yes, we showed that it converts alcohol to the acid.
So why would it stop at the alcohol in some tissues, or it doesn't?
It depends on the concentration. It depends on the abundance of CYP27A1. When it's highly abundant, it converts cholesterol to the acid. When it's not very abundant, it stops at the 27-hydroxycholesterol levels.
But there's no evidence that there's other enzymes to go from the alcohol to the acid. It's all C27A1.
It's all C27.
And it's also clear that the alcohol is formed and accumulates in tissues, or it always goes to the acid.
No, it can.
Some tissues will make the alcohol and stop.
Some tissues like lungs, they make acid preferentially.
Okay, so there's no other enzymes, and some tissues stop it out.
No, there is no other enzyme. All right, so hold on. Okay, if to simplify what is said on this slide is that CYP27A1 expression and its levels 27-hydroxycholesterol can be either beneficial or detrimental depending on the clinical situation. And this is exemplified by estrogen receptor positive breast cancer. And estrogen receptor positivity is extremely important. Why? So what studies found that in breast tumor cells, there is expression of CYP27A1 and 27-hydroxycholesterol. And in estrogen and in women of a reproductive age who have estrogen receptor positive breast cancer, high levels of CYP27A1 and 27-hydroxycholesterol are good, and it's a good prognostic markers independent of conventional breast cancer markers. However, when you become menopausal, the situation is reversed. And now high CYP27A1 expression and activity are detrimental, and perhaps CYP27A1 should be inhibited. In men with prostate cancer, the situation is more clear because CYP27A1 expression correlates, low CYP27A1 expression correlates with shorter disease-free survival and higher tumor grade. So with all this data plus knowledge that CYP27A1 inhibition could be detrimental in heterozygous CTX carriers, we undertook screening of the FDA-approved drugs for CYP27A1 inhibition. And we started with the screening in vitro enzyme assay. So, and what we have done, we screened approximately 157 compounds. Currently, there is approximately 1,600 FDA-approved drugs on the market. And of these 1,600 drugs, we eliminated all protein and peptide-based. We eliminated all controlled substances. And then we looked at the remaining, went through every drug on the market, and then it is called intuitive prediction, and then eliminated compounds with bulky structures, which we did not believe will go into the CYP27A1 active site. And then we ended up with these 157 compounds. We used E. coli expressed CYP27A1, and under the conditions of our enzyme assay, we determined how these compounds inhibit CYP27A1. And we used an arbitrary cutoff limit saying that we will only investigate drugs that inhibited to more than 75%, meaning that, there should be residual 25% of CYP27A1 activity. All right, we identified this compound. And then we determined the so-called inhibition constants or KI values for these compounds because, conditions of the enzyme assay determine what results you will get in your screening assay. And inhibition constants or KIs, it's an independent property of a compound for its inhibition of your enzyme of interest. So, and the lower KI value, the stronger the inhibitor is. So we identified all these KI values shown in this column, and then we classified all these inhibiting compounds. And micromolar, it's micromolar. Yes, it's a translation, so it's a micromolar KIs. So, and then we classified these compounds. Unfortunately, it was a different color here. So, and this says indication, and in parentheses, we indicated the drug target. Okay, so, and what you can clearly see on this slide, that the largest group of inhibiting compounds was among antihypertensive drugs. and not all of them, because there are at least 3 different types of hypertensive drugs among calcium channel blockers. And we have the whole spectrum from very strong inhibitor to a quite weak inhibitor. And for your information, when pharmaceutical companies develop different drugs, they, of course, do screening of their compounds for undesired or so-called off-target inhibition. And for them, the Ki value is a kind of a borderline limit. So if the compound has approximately 2 micromolar Ki value for some inhibition, usually it is considered as clinically irrelevant. All right, so what we got, the highest groups of, the largest group of compounds was among antihypertensive drugs. Then we found 2 quite strong HIV reverse transcriptase inhibitors. Then we found a bunch of anti-cancer agents, and 2 of them were prostate cancer agents, breast cancer agents, and then different tyrosine kinase inhibitors, and 1 non-sedative pain inhibitor. All right, this is all in vitro work. So the next question is whether these compounds inhibit CYP27A1 in vivo. And we investigated that by testing mice. So whenever the effect of any drug is determined by essentially 3 main parameters: drug dose, route of administration, and treatment duration. So, and what I have done, what I show on this slide is our treatment paradigms and drug doses, okay? And for comparison, I indicated the drug doses given to humans and normalize them per kilogram of body weight. And we assumed that an average human weighs approximately 70 kilograms or 155 pounds. All right, and we tested 3 compounds. It's a kind of a proof of concept study. So we used anastrozole. Anastrozole is an anti-breast cancer medication. So, and what we did, we treated mice for 7 days, and then we determined the levels of 27-hydroxycholesterol in plasma. And what these levels indicate, they indicate the whole body inhibition of CYP27A1, because when CYP27A1 is inhibited in different extrahepatic organs, then cholesterol is fluxed, 27-hydroxycholesterol is fluxed in the systemic circulation, and you see the whole body inhibition. So, and what we see that under our treatment conditions, we see a more than 50-fold, a 50% inhibition of CYP27A1. Then we also checked CYP27A1 inhibition in the liver. And we see that, yes, CYP27A1 is also inhibited in the liver, but to a lesser extent. Then we were wondering whether this inhibition affects cholesterol elimination from extrahepatic organs. And what we see, that inhibition of CYP27A1 does not affect cholesterol elimination either from the liver or extrahepatic organs. What does it mean? It simply reflects the fact that cholesterol 27-hydroxylation accounts for only 8% of cholesterol elimination from our body. The remaining 92%, they eliminated through bile acid biosynthesis in the liver. All right. Then we tested 2 antihypertensive drugs. And one of them, unfortunately, again, there is a conversion problem. One of them was felidipine, which is a strong CYP27A1 inhibitor, and the other was felidipine. Again, we treated mice, and then we determined the levels of 27-hydroxycholesterol in the plasma. And we see that a stronger inhibitor led to a higher inhibition of CYP27A1 in the whole body. Then we also checked inhibition in the liver and in the brain, and we did not see any difference between the 2 inhibitors. And then we checked cholesterol levels in the plasma, liver, and brain, and did not see any difference. Now I want, okay, so FDA approves, every year FDA approves new drugs. And it is impossible to check any newly approved drug for inhibition of CYP27A1. What people usually do in this situation, they look for a so-called inhibitory scaffold or chemical structure that can inhibit this enzyme. And this is what we have done. Here is, highlighted in yellow, all antihypertensive medications that inhibited CYP27A1. And in parentheses, you see the KI values. And what this slide, you don't need to be a medicinal chemist, because this slide clearly indicates that there is so-called 1,4-dehydroxypyridine scaffold. These are two benzyl rings, and then these two substitutions and these two positions. They're extremely important, and they determine how CYP27A1 is inhibited. If you make this substitution either shorter or bulkier, or this substitution, for example, is a little bit bulkier, your KI value goes up. Then so we have identified a scaffold here. Then if you look at two prostate cancer drugs, bicalutamide and abiraterone, you have a clear similarity to the structure of its substrate of CYP27A1 substrate cholesterol. When you look at anastrozole and fadrazole, we started to scratch our heads because we would never predict that these two compounds inhibit CYP27A1. And this is the reason why we do experiments. So these are two HIV, these are two tyrosine kinase inhibitors. And again, you see a similar structural fold. And the same you see with two HIV reverse transcriptase inhibitors. So now I want to switch gears and to talk about another enzyme, CYP46A1. So what our studies in mice showed that CYP27A1 is required to prevent cholesterol accumulation in the brain, because this metabolite, when it is its further conversion is blocked as a result of CYP27A1 deficiency. It is fluxed in the systemic circulation, then in the brain, and then in the brain it undergoes a series of reactions and converted to cholesterol, which is the major component of lesions. But what our studies showed, that both CYP27A1 and CYP46A1 can metabolize cholesterol in the brain, at least in mice. And that CYP46A1 is more efficient in metabolism of cholesterol than CYP27A1. And what does it mean? That when CYP27A1 is absent, perhaps increase in activity of CYP46A1 can compensate in part for missing CYP27A1 activity. And of course, both enzymes metabolize cholesterol, and metabolism by CYP46A1 is a preferential route of cholesterol elimination from the brain. So, and this slide just again, brief biochemical info on CYP46A1. This is a CNS-specific enzyme. It is expressed only in a certain neuron subtypes and initiates the major pathway of cholesterol removal from the brain. And since CYP46A1 is expressed in neurons, when you have neurodegenerative diseases, you have a loss of CYP46A1 in different brain regions, and there are disturbances in cholesterol homeostasis in the brain. And what I showed on this slide, that there are decreases in CYP46A1 expression and activity in many neurodegenerative diseases. Plus, we have a couple of non-neurodegenerative diseases like glioblastoma, which is an aggressive form of brain tumor. So what we have done, we did similar type of screening of FDA-approved drugs for CYP46A1 inhibition. Again, we used E. coli expressed CYP46A1. In this case, we screened more compounds, and we identified compounds that inhibited significantly CYP27A1 in our enzyme assay. And these are the compounds. And for them, we determined either the KI value or the apparent binding constant value. So, and perhaps we found, the largest group of compounds was among antidepressants, but the drug target was different. Plus, we have 2 antifungal agents. And again, we decided to find a scaffold, inhibitory scaffold. And here is the chemical structures of all these compounds. And you don't see any similarity. And we're wondering why. So what we have done next, we determined crystal structures of CYP46A1 with many of these compounds. So this is heme group, the site of catalysis, and the semi-transparent surfaces show the shape of the active site. And the drugs that bind compounds that inhibit CYP27A1 are shown in stick stick representation. And what you can clearly see in this situation, it is so-called in biochemistry term ligand-induced conformational fit. In a simple words, when some compounds bind to C46A1 active site, the active site changes its shape to accommodate these compounds. So, so we understood what is the difference for C46A1 inhibition. But perhaps the most exciting finding was not C46A1 inhibition, but C46A1 activation. Only 5% of drugs on the market activate the enzyme. And we found, at first we found in the in vitro enzyme assay that there is at least, a five-fold activation of C46A1 with a drug called, this is anti-HIV drug called efavirenz. And what we have done, we've done a lot of biochemical work, and we showed that this is allosteric inhibitor, meaning that it binds to the site on the surface of the enzyme. This is cartoon representation of the crystal structure of C46A1. And here is the enzyme active site buried inside the protein molecule. All right. So this is in vitro work. And of course, you need to show that what we have is relevant to in vivo. So we made this discovery of C46A1 activation approximately 10 years ago. And then we used a mouse model of Alzheimer's disease, the so-called 5-FAD mice. And we showed that low-dose efavirenz mitigates the manifestation of Alzheimer's disease in a mouse model. And then other people used our treatment paradigm and show the mitigation and very beneficial effects of C46A1 activation. This is in Parkinson's disease, Niemann-Pick type C1 disease, in certain types, not all types. Some seizures should be inhibited. In certain type of seizures, in acute depression, glioblastoma, and even in prion infected mice. And in parallel, people used gene therapy against C46A1, and they showed again very beneficial effects of increased C46A1 expression in Alzheimer's disease, Huntington's disease, Parkinson's disease, spinocerebellar ataxia, and amyotrophic lateral sclerosis. And when I sat down and analyzed the data from different diseases, I found that there are some common processes affected by C46A1 activation in different conditions. And this was common processes was decreased abnormal protein accumulation in different types of neurodegenerative diseases, improved memory and motor function, as well as changes in gene expression and protein phosphorylation. So, and in order to show that CYP27A1 is activated in humans, we did a small clinical trial with low dose efavirenz. It was only 5 patients because this trial was conducted during pandemics. And we started with 36 patients, but ended up with 5 patients. But what we showed that, yes, indeed, with 50 and 200 milligram of efavirenz, we activate C46A1 in the brain as indicated by plasma levels of 24-hydroxycholesterol. So these open bars are pre-treatment levels of 24-hydroxycholesterol, and then green bars or blue bar is increased 24-hydroxycholesterol levels at the end of the study. Then we discontinued the drug for 2 weeks, measured the plasma 24-hydroxycholesterol levels again, and see that, there is some decrease to the baseline level. So, and this is all I wanted to talk about today. So if to summarize what I said today, I believe that our data indicate that certain FDA-approved drugs can inhibit CYP27A1 and C46A1 in mice. However, we still do not know whether this is the case in humans, first. And second, if even there is inhibition of CYP27A1 in humans, whether it has any deleterious effects, because inhibition can be very strong, inhibition can be minor. And then it depends on the clinical context. So, but from a general precaution, perhaps particular attention should be given to some of the antihypertensive medication, calcium channel blockers. As I already said, there are two other groups of compounds of antihypertensive medication, and we checked some of these compounds, and they do not inhibit CYP27A1. So there are a number of options in terms of lowering your blood pressure. But as a general precaution, if there is any reason to believe that this particular compound can inhibit CYP27A1, perhaps it's not a bad idea to test baseline levels either in the plasma or in urine for specific CTX metabolites before the treatment and perhaps after 1 month of treatment, and to see whether there is some appearance of CTX-specific metabolites. Then our study suggests that perhaps when CYP27A1 is absent, another enzyme, C46A1, can compensate for lack of CYP27A1, but only in one organ, in the brain. So, and decrease cholesterol accumulation. And we also showed that CYP46A1 can be activated by low-dose efavirenz in mice and humans. And what our data suggests that perhaps it's worth considering low-dose efavirenz in a subset of CTX patients that do not respond well to chenodeoxycholic acid therapy, or they're resistant to this therapy. And, you know, it was a lot of work. The major contributors were Natalia Mast-Mori-Lam from my lab, and as well as funding. Thank you for your attention. Yes.
So can I have a question first, Mike? So I've been watching your very beautiful work over the last decade or so, and it's just really wonderful work. And I just, you know, this kind of brings up a point that Ken was talking about this morning in the session, that in carriers, there's 50% enzyme activity. You know, in CTX, there's a very small amount or deficient enzyme activity. And so, you know, I was fascinated by Dr. Piccoliva's paper showing that these routinely used drugs can inhibit, again, it's in the mouse model, you know, it's not in the human model, but can inhibit that enzyme activity. So, you know, I asked her to come and present that data. So I just wanted to make that point. So anyway, now my question.
So I noticed in your assays for the CYP27A1, you're using adrenodoxin as the reductase.
No, it's both adrenodoxin and adrenodoxin reductase, which are redox partner for this mitochondrial
Right. Is that the only, so adrenodoxin is the iron sulfur protein?
Yes, it's an iron sulfur protein.
So those two proteins, are they the only proteins talking to CYP27A1?
Yes, yes.
This is clear.
Yeah, it's crystal clear. It's not a microsomal P450. It's only two.
Are these the physiologically
relevant? Yeah, the physiological.
How do you know that?
Yeah, because any CYP should require electrons for their enzyme activity.
No, but how do you know it's adrenodoxin reductase?
It has been established centuries ago.
Okay, because there's many P450 reductases.
Yeah, but these are this is mitochondrial P450. It resides in the inner mitochondrial membrane, and there are 57 P450.
Okay, yeah. Okay, so this is clear. Is there any possibility that your FDA drugs are inhibiting the reductase or the adrenodoxin?
No.
Are they binding to the heme? Do you see spectral shift?
Yes, we see, and you know, I showed this shift as
very nice. Okay.
And I don't know if I clarified that very well, but I meant what Ken said, you know, that something stressed the pathway. So in carriers, you know, they showed back in the 80s that taking a drug like that binds bile acid can stress the pathway. So the metabolites that you normally see in CTX would rise in carriers. So.
Or it could be cholesterol, but fascinating work. That's a massive amount of work, actually. Congratulations on that. But in the case of CTX, of course, you wouldn't want to inhibit CYP27A1 because,
yes,
I mean, basically you don't have CYP27A1. But I can think of a situation where it might be useful to have an inhibitor of CYP27A1, and that's in the 7-alpha oxysterol 7-alpha hydroxylase, the CYP7B1 defect, which is, of course, really a fatal condition. These kids live not very long. And cholic acid doesn't suppress that pathway because that occurs in the alternative pathway. You know, cholesterol gets converted to 27-hydroxycholesterol, and then, as you showed, it goes through to cholestanoic acid, the monohydroxylated 3-beta-delta-5. Now, that is the most toxic bile acid to the liver, and the next step on is to 7-hydroxylate that, and that's the CYP7B1
Right.
that actually deactivates the toxicity of that monohydroxy bile acid. So patients that lack CYP7B1, the oxysterol deficiency, they accumulate massive amounts of cholestanoic acids, and that's quickly destroys the liver. And as I said, colic acid didn't work. In fact, you know, the few cases we've tried it in, it failed. What I was looking for on the very first case that when we first described the oxysterol 7-alpha-hydroxylase deficiency was, could we find an inhibitor of CYP27A1? Because you want to block that pathway completely to stop the production of monohydroxylated bile acids. So I was interested in some of those drugs because, you know, it's curious, maybe selecting one or two of those in the future when we see the next patient might. But as I recall, cyclosporin is an inhibitor of,
Yes,
isn't it? I didn't
yes.
see that on your listing, but
it's a very weak inhibitor
it's a weak inhibitor
for CYP27,
relative to the others.
relative to the others.
Okay, that's useful to know.
And then we don't test non-inhibitors. We want to identify new inhibitors.
Yeah, and then the other issue with the CYP46 enzyme is that, you know, when you generate these 24-hydroxy cholesterols, they're potent repressors of CYP7A1. So they would in the liver, you know, suppress cholesterol synthesis. So.
But it accounts for only 8 to 9 milligram of cholesterol elimination from the brain. So it's not of, yes, it suppresses, but.
It's more important in the brain.
Right. Mm-hmm. But it's a great point, and I never thought about, you know, in deficiency of CYP7B1, that suppression of CYP27A1 could be beneficial.
Yeah.
It's a great point. Mm-hmm.
Yeah. This is why, in fact, the alternative pathway which leads to chemo is much more important in the neonatal period, the fetal and neonatal period, because they actually have reduced activity of CYP7A1, and it's thought the reason for that is the alternative pathway is active, generating oxysterols which suppress CYP7A1. So bile acids are not made until many weeks or months after birth by the classical pathway, because in adults this pathway is relatively minor. That's been very well proven over the years. You know, going back to Reno Blachowicz work in the, I don't know, the mid-late 70s. So, but the neonatal period is a more fascinating period from the point of view of the discrimination of these two pathways. And CYP27A1 is really important in early, but not so important
Later. Yeah.
Very nice.
We're running a bit behind, so I think we're fine to run on a little bit after the meeting, Jean, aren't we?
I will check, but I think so. Yeah.
Okay, so
Good. Quick question, and beautiful talk. I was curious about this association of CYP27A1 with breast cancer. Do you have any concerns around potential for gene replacement bringing about a risk for oncogenesis, or do you think this is just a postmenopausal problem?
I think this is postmenopausal thing. So, and it's good to have normal CYP27A1 activity. First, you have to survive without breast cancer during your reproductive age. And if you get to premenopausal, then you start worrying about all the rest. But at first, you should not get cancer during your, you know, reproductive age. This is my philosophy. This is how I understand things. You worry about menopause later.
Yeah, it's just fascinating, the association with the breast cancer. And, you know, they say that to be a carrier for something, it has to give some sort of protective effect. So it kind of makes you wonder if that's you know, what's happening there. So any questions online or?
Question. This question is from Dr. Gonzalez. CYP27A1 transgenic expression could have an anti-mural effect? Question mark.
Transgenic, but why it's the question to me? If we are talking about transgenic
expression could have anti-moral effect. It came up during your presentation, so I don't know.
But
tumoral? Yeah.
Tumoral.
Antitumoral, yeah. Anti-cancer. Anti-cancer is what Dr. Gonzalez is saying.
Well, I think we'll move on to the.
Yeah, it depends.
Okay.
Thank you. Fantastic. And we'll have our next speaker start here, Dr. Ling.
Here, I think this is a better microphone.
Yeah, I would take that.
All right. Okay. I am Leiling from NGM and very happy to get invited to this session. And it's an honor and a pleasure to speak here. So the next slide is this one.
Green arrow.
Okay. Yeah, here's my disclosure. And this one? Oh, this one. Okay. All right. This is this big one. All right. Okay. So this audience know everything about the CTX. So I will just go through it quickly. The mutation, as a consequence of the mutation in CYP27A1, not only do patients lack primary bile acids, they also have the accumulations of the toxic intermediates, the cholesterols, the bioalcohols. Most of the time, we think about supplement the missing bile acids, but not too much has been done on suppressing those toxic intermediates from happening in the first place. We learn it from the experts and patients, the symptoms, the devastation, the CTX patients experience, the neonatal cholestasis, cataracts, diarrhea, xanthomas, neurological symptoms, and we're with you. And the prevalence is pretty low in general population, but can be quite high in certain population. I learned from Dr. Falik here that the 1 in 11 carrier frequency in certain Druze population, that's quite high. And also in juvenile cataracts patients, the prevalence is also pretty high. There is no officially FDA approved therapy, but that may change quickly with your NDA. Okay. I wanted to talk a little bit more stress about this bile acid intermediate, the 7-hydroxy4 cholesterol, which is a long name, 7-alpha-C4. I would just call it C4 for simplicity. Although we all know that the cholesterol and bioalcohol are the gold standard, one of the gold standard of diagnosis, Dr. DeBarber's work back in 2010 has told us that the C4 can have very important utility. And especially her recent work in the Journal of Clinical Lipidology, as well as posters at this meeting, that C4 have improved utility than cholesterol because they are patients, CTX patients with normal levels of cholesterol. So we know the CDCA can inhibit C4, but that suppression is not optimal. It's not totally normalized in patients. So as a consequence, some patients, they still have this elevated C4. The toxic intermediates still form, and we still need something more to address that. And what I am going to talk about is we learned there are quite a few molecules that potentially be useful, and that's a good sign. And this particular one would be inhibiting CYP7A1 and hopefully to reverse the accumulation of toxic intermediates. So bile acids are very paradoxical molecules. You can't live without it, but you cannot live with too much of it. In humans, we have evolved this mechanism. It's a feedback mechanism that to automatically tone down the accumulation of hepatic bile acids. And this is done through this hormone called FGF19. FGF19 is an endocrine hormone that's secreted from the ileum to act on the liver to suppress the classic pathway of bile acid synthesis. And it is part of our physiological regulation of bile acid metabolism in the gut-liver axis, the enterohepatic circulation of bile acids. This FGF19 FXR pathway actually has utility in multiple indications, particularly in chronic liver disease. That's where we did most of our trials in. And this analog that I'm going to talk about, aldafermin, I'm just so impressed that Dr. DeBaba pronounced it perfectly, because a lot of people cannot pronounce those names. We actually develop it in multiple disease indication. So aldafermin is the first and only FGF19 analog currently in clinical development. And it has the pharmacological characteristics of a biased ligand or a selective modulator. As a result, it retains the metabolic activity of FGF19. It is non-tumorigenic in multiple animal models with even a prolonged exposure. And it further blocks FGF19-induced tumorigenicity. Mechanistically, aldafermin works through the FGFR4 beta-closal receptor on hepatocyte to suppress CYP7A1. It also works on FGFR1C beta-closal in adipose and neurons to regulate glucose metabolism, insulin sensitivity, and energy expenditure. So aldafermin impacts several key pathways in disease pathogenesis to reverse metabolic activity, metabolic toxicity, and reverse especially bile acid toxicity. I want to show you some of the data that we have in animal models in cholestasis. Cholestasis is characterized by the impaired bile flow and then the accumulations of toxic bile acids in the liver, and which leads to inflammation and fibrosis and ultimately cirrhosis. And it's actually, in humans, is one of the most debilitating chronic liver disease. So in this MDR2 knockout mouse model, which recapitulate the human cholestatic liver disease, PSC and PFIC. In this model, treatment with aldafermin, we saw that it can effectively decrease liver bile acid pool and serum bile acids. It stopped liver inflammation. It reversed liver fibrosis and improved biliary health. BDL model is bile duct ligation. It mimics the human, in human disease, the biliary obstruction. And a NIT model is a chemical-induced cholestasis model. In both models, we saw aldafermin reduce liver enzymes and reduce hepatic retention of toxic bile acids. So the aldafermin clinical development program, we actually have multiple indications. Cholestasis, non-alcoholic steatohepatitis, and bile acid diarrhea. So over 1,000 participants have enrolled in aldafermin trials, and over 800 of them have received aldafermin treatment for up to 1 year. And we have conducted and completed phase 2 trials in primary sclerosing cholangitis, PSC, primary biliary cholangitis, PBC, non-alcoholic steatohepatitis, NASH, and bile acid diarrhea. I will tell you what we have learned from these trials in the next few slides. So the first up is PSC, primary sclerosing cholangitis. It is a human chronic cholestatic disease characterized by the inflammation and fibrosis of bile duct, and which leads to strictures and complications. And there is a toxic bile theory in PSC that this regurgitation of bile through the leaky bile duct actually cause liver injury and inflammation, and then leads to the cholangitis and fibrosis. In this 12-week double-blind placebo-controlled trial, we treated 62 PSC patients with aldafermin 1 milligram, 3 milligram, and placebo. And we saw dose-dependent reduction in fibrosis markers and liver enzymes, ALT and AST, although we did not see any change in alkaline phosphatase. Why ELF? ELF is a fibrosis marker. I love to see what you talk about, the surrogate endpoint, potentially reasonably likely to predict the clinical benefit. This is one of the candidates. It is blood-based. It can be measured non-invasively without the need of liver biopsy. And most importantly, in PSC, ELF is a strong predictor of disease outcome. The higher the ELF, the worse the prognosis. And in this 12-week trial, aldafermin arm, both aldafermin arm were able to reduce ELF about 0.3 to 0.4, comparing with an increase about 0.1 with placebo. The most pronounced effect actually seen in patients with more advanced disease, those with baseline ELF more than 9.8. And in those patients, we saw a reduction of 0.5 to 0.6 with aldafermin. PRO-C3 is another marker, fibrosis marker, non-invasive, blood-based. And like ELF, it is also prognostic in PSC. The higher the PRO-C3, the worse the disease outcome. And we saw aldafermin able to lower PRO-C3 by 30% to 40%. PBC is a sister disease for PSC, occurring 1 in 1,000 women over 40. And in this 4-week phase 2 trial, we tested aldafermin 0.3 milligram and 3 milligram versus placebo, and saw dose-dependent reduction in liver enzymes in ALP, in ALT, and AST. And we also saw reduction in IgM and IgG, and a trend of improvement in pruritus in itch. The majority of the aldafermin database actually come from patients in NASH trial, recently renamed to MASH. We actually tested aldafermin in more than 400 non-cirrhotic NASH patients. This is done through multiple phase 2 trials. And we saw dose-dependent reduction in multiple parameters. So here it is about 50% to 60% reduction in liver fat, 40% to 50% reduction in ALT, 40% reduction in AST, and a higher proportion of patients on aldafermin arm achieved the histologic endpoint, which is done by paired biopsy. That is required in NASH. More of the patients achieved fibrosis improvement of at least 1 stage with no worsening of NASH, as well as NASH resolution with no worsening of fibrosis. So overall, in NASH non-cirrhotic patients, the biochemical, the imaging, and the histological endpoints all aligned to show that this molecule is having a benefit in this patient population. We also done a separate NASH trial in patients with compensated cirrhosis. And this is a 48-week study with aldafermin 0.3 milligram, 1 milligram, and 3 milligram comparing with placebo in 160 cirrhotic patients. And we saw dose-dependent reduction in non-invasive markers, and especially here, again, ELF, the enhanced liver fibrosis. Like in PSC, ELF is also prognostic in NASH and has been recently approved by the FDA to predict liver-related events in NASH with compensated cirrhosis. In this 48-week study, we saw dose-dependent reduction in ELF in both autophamin arms, and especially the primary endpoint, because it's the first time the ELF is used as a primary endpoint in NASH. And we met the primary endpoint with a p-value of 0.0003 for the autophamin 3 milligram arm versus placebo. And the ALT and AST reduction was significant almost at every time point that we assessed. The gray line is a placebo, the blue line is the 1 milligram, and the green line is the 3 milligram. And this is for same for reduction for both ALT and AST. And also more patients, this should be 20%, the numbers should be corrected. More patients on autophamin achieve the histological endpoint of fibrosis improvement of at least one stage. So the reason I go through all of these trials is to show you this slide. All the slides previously are disease-specific markers or fibrosis markers or liver injury markers. This slide is on C4. In all of the trials, autophamin trials conducted to date, whether it's patients with PBC, PSC, NASH, diarrhea, or healthy volunteers, there is one constant that we see across all of the trial, which is a C4 inhibition. And here in PSC, this is actually 90% inhibition, suppression in C4. In PBC, 94% reduction in C4. In NASH, it is 93% reduction in C4. And in bile acid diarrhea, here the data is from individual patients, and you do not need a statistician to tell you that this is statistically significant. So although we have never done any study in CTX, that's the acknowledgement we need to make. We have no data in CTX, but based on the data from all the autophamin trials conducted to date in multiple indications, we think the molecule should inhibit C4 and hopefully stop the accumulation of toxic intermediates in CTX patients. So in summary, autophamin is a first-in-class engineered analog of the human hormone FGF19 in clinical development. The program had multiple clinical data sets across multiple indications. The constant from all of these trials, totally different etiology, revealed one thing that is in common, that is anti-fibrotic and inflammatory, and most importantly, the very significant reduction in C4, as well as serum bile acids. So autophamin has quite a large safety database, and in over 800 patients with more than 1 year of with up to 1 year of exposure, and it has been found to be safe and generally well tolerated, and the most frequent adverse events were mild gastrointestinal symptoms. That we hope that autophamin with a novel mechanism of action by targeting this upstream node of disease pathogenesis at C4, at CYP7A1, may provide a treatment option for patients with CTX. We have to stress that this molecule is not a bile acid replacement therapy. You need bile acid replacement therapy. This is on top of potentially as an add-on for patients who are not optimally controlled with bile acid replacement therapy. And I'd like to thank all the patients who contributed to all of the study, more than 1,000 of them who participated in the study, as well as the investigators, coordinators, because it is a huge amount of work for the patients, for families, for investigators, for coordinators, for site staff to participate in any clinical trial. And we thank them from the bottom of our heart. Yeah. So I will stop here and love to take any questions.
And as we have discussed throughout the meeting, we all know that chenodeoxycholic acid is a standard of care in CTX, and, has been used for the last 50 years to basically decrease those toxic intermediates. But there is still a subset of CTX patients that, maybe it's because they're treated later or the different reasons that people have hypothesized, but that still continue to deteriorate a small portion. So, we're just thinking of ways to expand the toolbox and the things that are available. So, aldafermin seems to be a great way to decrease that pathway and to completely shut down the pathway. The biochemical data is very convincing. So I appreciate you coming and giving a talk on that. Ken has a question.
Just a quick one, because I worked with NGM in the early phases, as you know, and I can't remember the number, NGN something a number it was.
282.
282, yeah. And as I recall, its route of administration was IV. Is that true for this one?
It is a sub-Q. Patients
Sub-Q.
self-administer.
Yeah.
We do the
Right.
study in NASH. They self-administer.
Yeah, which is sort of less attractive in terms
Yes.
of long-term therapy. But and then the other thing I recall was that loose stools and diarrhea was an issue in the early work that we published with NGM.
You have great memory. Loose
Well,
stool
some
and
people tell me I
diarrhea
don't.
was the issue in earlier studies because
My
we
wife.
tested too high a dose. Later on, we find that you have to fine-tune the dose. With the lower dose, you can achieve effective suppression of CYP7-1 without the diarrhea, the loose stool, because that part actually, Dr. Camilleri from Mayo Clinic did a beautiful study. It shows that at a higher dose of aldafermin, it stimulates GI transit.
Yeah, that's good. Very nice. I mean, I think the application for fibrosis will be amazing if that pans out.
Thank you.
Oh, sorry.
I'm just curious what indications you're pursuing for pivotal trials and what's the way forward for y'all, just briefly.
Excellent question. I am sure that anyone who works in biotech or pharmaceutical companies, they have to go through very stringent internal evaluation. It's look at the unmet need, look at the potential molecule, potential competition, et cetera, et cetera. Currently, our company, we're planning to do a pivotal trial in primary sclerosis and cholangitis, which is also an often rare disease. But the patient population is slightly higher than the CTX.
Any questions online, Jane?
Question from Sue Stewart online. Could any of the patients in the chronic diarrhea group have been undiagnosed CTX?
Wow, that's a great question. I never thought about that. We should get Dr. DeBarber involved, right? Because those patients, they never know anything wrong, right? It's just a symptom. Yeah, because some of those, the C4 is like 200 nanogram per ml. Yeah.
Yeah, well, I think it's definitely an underappreciated symptom, the chronic diarrhea, that most people don't recognize. So could be. But I think we'll take a very quick break now, just for 5 minutes, everyone to have a vibrate cup of coffee, and then we'll start up with the second session. So thanks very much, everyone. Okay, so our next speaker is Dr. Zipporah Felix-Akai. We're very grateful to have her here all the way from Israel. So she'll be talking about real-world data supporting the use of CDCA treatment in pregnancy in CTX. Thank you. Go back.
Okay, so good afternoon, and thank you for the organizing committee who invited me to give this talk today. I would like to thank my team, all the collaborators from Israel and all over the world. I would like to thank a lot to the CTX Alliance and to the ULF Foundation for supporting my trip here, and it's a great honor, and I'm very happy I can be here personal and give you this talk. Thank you. And I'm a volunteer member of the board. So you saw these pictures before. You were in the same conference in Jerusalem. And again, I would like to dedicate my talk to Professor Berginer, who really pioneered the field of CTX in Israel. And he changed the frame of the disease and turned it to be from a lethal disease to a treatable disease. And this is a huge thing. So what I'm going to talk today is about a very practical issue. We are now reaching the area that we have CTX patients who are fertile, getting married, and want to have children. And they are very concerned about the question whether to be treated or not to be treated during pregnancy. Will it harm the fetus? Will it, if they continue the pregnancy? Will it harm them if they discontinue the treatment? So that is what the subject of my talk. So, you know, 5 lines of introduction: CTX, B allelic pathogenic variant in CYP27A1. Deficiency result in impaired production of the primary bile acid, especially CDCA, and treatment with CDCA implies functional cure. It means that it halts the clinical CTX progression usually and corrects the biochemical abnormalities. When, as I said, when women with CTX are recommended usually to stop treatment. And why is that? Because there is a theoretical risk, and emphasize the theoretical risk to the fetus, but both the patients and the clinicians are very concerned about stopping the treatment. Dr. Barduel recently published a paper in the Journal of Clinical Lipidology, and he wrote about the concerns that he has as a physician who treats CTX patients and his patients regarding stopping the CDCA treatment. The reassurance from the CTX specialist was the primary factor in these women's decision to continue treatment during pregnancy. So to date, CDCA is designated by the FDA as pregnancy category X with a warning that it may cause harm to the fetus. I would like to suggest that this designation should be revisited considering the animal studies that were done and lack of human data regarding treatment during pregnancy. The designation is based on non-CTX animal studies and very high doses of CDCA that were given to those animals. And all this may not be relevant at all to human with CTX and pregnancy. So if you look today at the product information of cannabidiol, for example, from Nextgene Pharma, it says cannabidiol is contraindicated in women who are pregnant or may become pregnant. There are no available human data regarding the use of cannabidiol during pregnancy. However, cannabidiol may cause fetal harm when administered to pregnant women. On one side, there are no data. On the other side, they say it may cause harm. Why do they say that? Where does it come from? Another cannabidiol from Briggs drugs in pregnancy and lactation. Pregnancy recommendation contraindicated. No reports on the use of cannabidiol during human pregnancy have been located. So no reports, but still contraindicated. Based on the observed hepatotoxicity of this agent, the use of cannabidiol is contraindicated during pregnancy. So what is the hepatotoxicity that they are talking about? There were studies showing that in several animals, there were serious hepatic, renal, and adrenal lesions, hemorrhagic necrosis, okay? In offspring of female rhesus monkey that were given cannabidiol 60 to 90 milligram per kilogram, which is 4 to 6 times the human dose, the recommended human dose, from day 21 to 45 in pregnancy. Another study showed that neonatal baboons developed hepatic lesions following maternal administration of cannabidiol 1 to 2 times of the human recommended dose throughout the pregnancy. Biochemically, the main hepatotoxic property of cannabidiol in animal is due to the principal metabolite, litocholic acid. Now, in human, litocholic acid is readily metabolized to a poorly observed non-toxic compounds. Animals like rabbit, rhesus, monkey, baboons are unable to fully form the sulfate conjugate to detoxify the metabolite, and thus they are susceptible to liver damage. But in human, because we know how to detoxify the litocholic acid, there is no problem. So, but all this designation was based on the animal studies. Cannabidiol was proven to be not teratogenic in animals. No fetal malformation were observed. No evidence for fetal abnormalities in rats, hamsters, mice, and baboons during reproduction studies. Embryo lethality was observed in rats after administration of a very high dose, 8 times the human recommended dose of cannabidiol. The drug was not carcinogenic in mice and rats when those were like those used in human. Higher dose were carcinogenic. So based on all those studies that I showed you, the FDA designation is based actually on animal studies which are not affected with CTX and have a different metabolism of CDCA. The lack of human data regarding the safety of CDCA treatment for the developing fetus has caused uncertainty for pregnant women with CTX and their healthcare providers. So both the women and the physicians are somewhat confused. So what do we know from human data about CTCA treatment during pregnancy? Berginer and colleagues reported for the first time in 1981, 6 patients. They were of a Moroccan Jewish origin. Between them, there was one woman who gave birth to a newborn with high plasma cholesterol, 18 microgram per ml, and cataracts at birth, congenital cataract. Concentration of cholesterol in the baby were normalized after 6 months. Finding, what do we learn from this case? First of all, this was the era before we know about the CDCA treatment. The first paper about the treatment was in 1984, the New England Journal of Medicine by Berginer and Salin. And since then, CTX patients started to be treated. So here, there was no treatment at all. Of course, the woman was not treated. And she had a very sick baby. He was born as if he is affected with CTX, although we know he must be heterozygous to the mutation, but of course not didn't have CTX himself. The other thing that we can learn is that cholesterol is transferred through the placenta from the mother to the fetus. Thus, it causes some harmful environment to the growing fetus. A second paper by Berginer and his colleague several years later, in 1988, after treatment was initiated, he recommended, he says, withdrawal of CDCA treatment during pregnancy could lead to neurological deterioration of the mother and also to accumulation of cholesterol and C27 bialcohols in the developing fetus and stillbirths. He saw stillbirths in 2 pregnancies. There were also 2 neonates that were born prematurely and showed evidence of potential intrauterine growth retardation. And this is in women that were not treated during the pregnancy. So Berginer and his colleagues say, we hypothesize that CDCA treatment in pregnancy enables a better outcome to the affected mother and their fetuses. And they say in the end of the paper, further evaluation is needed to pregnancies and offspring of CTX mother to confirm or dispel this hypothesis. So this was in 1988. Since then, there were no papers published about CTX patients and treatment during pregnancy. So we decided to assess the safety of CDCA treatment during pregnancy. I must say that I'm coming from Israel. We have a very high prevalence of CTX in specific populations. And we know to diagnose the patients very early. You said today 18 months. We know about them in pregnancy. We identify couples at risk. And immediately when they give birth, we test the baby. Genetically, we know the founder mutations in those populations. So we diagnose them very early and treat them. So we have many young women that want to get pregnant. They are affected, they are treated, they are healthy. As I said, they do everything. And the question of treating or not treating them during the pregnancy is a very practical issue. So we conducted a retrospective study of our patients and patients of our collaborators from all over the world, 9 mothers, 19 pregnancies. And we would like to suggest that CDCA treatment during pregnancy may be safe for the developing fetus and that the benefit to both mother and fetus is outweighed the potential risks to the fetus from the CDCA exposure. So each co-author provided a narrative clinical summary. So we have all the summaries of the 9 mothers with CTX that were either treated or not treated with CDCA. For each mother, we report the CDCA treatment status before and during pregnancy, complication, experience, and the health of the newborn, including follow-up into childhood and adult. So, as I said, 9 women, 19 pregnancies. There were no side effects in any woman for the pregnant women that were treated during pregnancy. The 11 pregnancies where mother continued CDCA treatment, no complication, offspring was born at or near full term with normal birth weight and normal upper scores. In 8 pregnancies, where no treatment was given, 2 experienced elevated bilirubin in newborns soon after birth. 1 offspring showed mixed expressive receptive language development disorder that resolved. 1 woman that stopped the treatment deteriorated neurologically irreversibly. Her SARA score went from 1.5 to 4. 5 females and 14 males were born. No medical issues were present among the 19 offspring. On follow-up, they were all reported to thrive well and present normal development. There is no evidence for hepatotoxicity or teratogenicity of CDCA treatment during pregnancy in our cohort. Follow-up of those children ranged from 4 months to 25 years, all present normal growth and development. There were no medical problems, and they all went to formal, regular, and academic education. Information was not available for one of the 19. So I would like to discuss the question, how should we follow and treat pregnant women affected with CTX? And what is the best way to protect both the mother and the fetus? So full data from Berginer et al. Showed intellectual disability in 2 of 18 offspring from 9 untreated mothers with CTX, while 11 offspring of 7 fathers with CTX showed no abnormalities. Information regarding fetal loss was not available for those families nor for siblings of the patients. Previous reports of pregnancies in mother with untreated CTX have shown that complications, including fetal and infant death, appear to be more common. Of 10 pregnancies in 4 untreated CTX mother, Yahalom and his colleague reported 2 miscarriages in the first trimester and 1 term stillbirth. This data suggests that untreated CTX women may create an abnormal intrauterine environment for the developing fetus. Possible placenta transfer of elevated cholesterol and other CTX-associated metabolites like C27 bile alcohols may occur from the mother to the fetus, thus harming the developing fetus. Previous studies suggested that CTX heterozygous fetuses are continuously exposed to abnormal levels of bile alcohols, cholesterol, and apolipoprotein B, whose deleterious effects on the adult brain were documented by Seeline in 1987. This is a very similar situation to a disease called phenylketonuria. High levels of phenylalanine in pregnant women were shown to be harmful to the fetal growth and development, while treatment of maternal PKU resulted in normal outcome of their pregnancies. If we keep those women on normal phenylalanine levels, i.e. they keep a very strict diet that helps them to keep the normal phenylalanine levels, the fetus is not exposed to high levels of phenylalanine and is developing normal. So the fetal environment is extremely important to the development of the fetus. It is recommended that phenylalanine is monitored and maintained throughout pregnancy. In our cohort, mothers with CTX that received CDCA, the maternal plasma cholesterol levels were generally maintained at either normal or slightly high levels, 8 to 10 milligram per ml. We did not measure plasma cholesterol in newborns at birth. Recently, there was a publication from a modified Delphi study, like experts' opinion on different issues regarding CTX. And the question of treating or not treating women during pregnancy was asked. And there was 67% agreement and no disagreement with the following sentence. Research indicates that treating CTX mother with CDCA during pregnancy is an important means of protection against damage to the fetus and miscarriage. Despite the agreement among the most experts, consensus was not reached. So this is reflective of the limited data that we have on humans, on pregnancies. So, you know, the physicians do not really know because we really lack data. So these are all the collaborators that participated in the study, and what did we conclude? The data we present support the benefit of continued CTCA treatment in pregnant women with CTX for both the affected women and for their offspring. More studies of pregnant women with CTX and their offspring are needed before consensus regarding the safety of CTCA exposure for the developing fetus may be reached. The real data, the real world data that are presented here will help physicians and patients make informed decisions about CTCA use during pregnancy in women with CTX. But I must say that our data suggests that the existing contraindication for use of CTCA in pregnancy in women with CTCA should be revisited. So this work was recently published in Genetics in Medicine, and I would like to say again, all my collaborators and the participating women from all over the world. Thank you. To answer.
Yeah, maybe I missed it, but did you do the genetics on the 19 offspring? Were they
Yeah,
all
yeah.
they were all normal?
All no,
So there was no heterozygous in the
no, the children, the newborns,
children, yeah.
they are all obligatory
Okay.
heterozygous because the mothers.
Oh, okay.
are
I'm sorry, I
sick.
missed that then. Yeah.
Yeah.
And then the other thing I wanted to mention was, you know, when one thinks about it makes comparison between doses of exposure in animals being really high, and people have a habit of doing that, and they say, well, it's so much higher than we would give in a human. But you have to take into account that when you do an animal study with a drug, you have to account for allometric scaling. And so because these animals, you know, have a much higher metabolic rate, for example, if you're working with mice or rodents in particular, you know, you would have to factor in about somewhere between a 6 and 8 time higher dose to be equivalent to what it would be in a human. I'm not sure what it is for rhesus monkey, but I think it is higher. So when you see a high dose, there's always a tendency to say, well, you know, we never use that dose in humans, but you have to, you know, do that mental calculation, and that's why you use higher doses for that reason. But
I understand, but there is still the biochemical pathway, which is different between the animals and humans.
yeah, I mean, I still say that chemo is toxic to the liver. I mean, it becomes toxic when you don't have bile flow. And because then you accumulate a lot of chemo in the hepatocytes, and chemo is very destructive to the mucosa and to hepatocytes. So, but you know, it's not destructive if everything bile is flowing, because you clear it, you're continually clearing it. And this is why really, you know, in the early studies of using chemo for gallstone dissolution, one of the side effects in a significant number of the patients was elevation in transaminases. And part of the reason was in those patients, they have normal synthesis rates of colic and chemo, and then you pile in more chemo on top. So now the liver is seeing a really high amount of chemo,
But
and that
those
begins to
women-
account for the toxicity. It is at the end, they are very hydrophobic bile acids. So
We follow those women very carefully. All the liver
Yeah.
enzymes were normal during the whole pregnancy,
yeah.
so there was no problem.
Yeah, I might also say that
But-
we've had 2 patients that with bile acid synthesis defects, not CTX, that actually they've been on colic acid for many, many years, got pregnant and continued. They didn't stop colic acid because they were pregnant. And they gave, you know, they gave normal deliveries, and there was, you know, no side effects or anything with chemo.
That's
Just
great.
anec-
That's
just
important
anecdotal. We've
information.
not written it up.
You
Yeah.
should publish it.
Yeah.
You should publish it, Ken. You should.
So I had a comment and a question. You had the limitation there, or the caveat or suggestion that more research is needed on pregnant women, but at the same time, maybe we've done enough. Maybe you're already convinced that it is safe during pregnancy and it's not safe to stop it during pregnancy. I'd love to hear your thoughts on that.
I'm pretty sure that it's very safe. And I think that every woman should be treated. And I think that the harm that can be caused because of stopping the treatment is unreversible. it doesn't worth it. So I think it's very, very safe.
I
But
agree.
I don't know, like for instance, you guys, maybe there are in the audience here, know how should we convince the FDA to change the category X,
I think we should
which is
start.
very, very important.
I think we should start that conversation with them.
Yeah.
Conditions so rare, and then to have the situation where you're then looking for one of the patients to get pregnant is even more rare. So you're never going to get a sample size that
Right.
I mean, we can continue to collect data, and we should have a registry, that sort of thing. But
Right.
but we should also maybe be careful about our language, not to suggest that we're still uncertain. That I think a lot of us are comfortable with it.
Yeah.
And then my
Thank you.
question came up before Ken mentioned. I was going to ask about colic acid. Do you know what the pregnancy determination is by the FDA for colic acid? Is that category X? But do you know of any other data other than the one that you mentioned? Okay.
Do we have any online questions? Okay, so I was a bit remiss. I was supposed to gather everyone together for a photo before we went out to the break. Especially if we're going to run on a little bit longer, Jean, should we do the photo now quickly? So just to make sure that we get everyone. So if everyone wouldn't mind coming up to the front, and we'll just take a quick group photo. And many thanks again. That was a great talk.
Okay, well, thank you. It's really good to be here. I really like these small meetings, I must say, much better than the big ones. But anyway, I have to admit, I don't have a lot of new stuff. And, from last year, it's difficult to acquire new data in 1 year. But so a lot of these slides you will have seen. I should start actually by way of transparency to point out that I do have financial disclosures, and one of them is with Asclepion Pharmaceuticals, because that was the company I founded or co-founded when we invented colic acid monotherapy, and we brought that to FDA approval in 2005, and it is approved for CTX. So anyway, let me start by just looking here at I don't do newborn screening. I want to stress that, but I do diagnose and pick up infants and neonates that have CTX, primarily because of this observation that we made in the year 2000. We were the first to show that a transient neonatal cholestasis can be a feature of early stage CTX, and in many cases it resolves and disappears, and I think that from that perspective it may be why some patients are not picked up, because it's not in the neonatal screen. So anyway, you see here the average age at diagnosis, as you know, is been quoted as around 35 years. So and I'm going to focus a little bit on the pathophysiology, because if you look at the major phenotype of CTX, what you have is, I've just said, neonatal. We believe really what we believe about the neonatal cholestasis is it's most likely secondary to the reduced bile acid pool size that is a feature of all newborns, including healthy newborns. And the potential cholestatic effects of the buildup of bile alcohols in a period where the neonate is particularly susceptible, and particularly if there's a stress situation, which we were discussing earlier, things like TPN, induced cholestasis, would be one particular stress. The diarrhea, again, I think can be attributed to the small bile acid pool size with an intraluminal bile acid concentration that is below the critical micellar concentration, the CMC, for effective lipid absorption. And also, we proposed some years ago that it was possibly due to the cathartic effects of bile alcohols. I not going to discuss cataracts, so you're all aware of that. It's considered to be due to the accumulation of the cholesterol and the CNS effects as well from cholesterol. But I think that the most important thing is that to note that I think CTX most likely presents early as a transient cholestasis that is usually resolved. And then those patients go on to be relatively asymptomatic until much later in life. So just to summarize, I've simplified the bile acid pathway to show you here. This is cholesterol. It's a complex pathway of 17 enzymes that are responsible for converting cholesterol into the primary bile acids of cholic and chenodeoxycholic acid here. And many changes occur to both nucleus of cholesterol and the side chain. But the critical one is 27-hydroxylation. The purpose of introducing that 27-hydroxyl is that is the initiating factor for reducing the carbon 8 side chain of a sterol to a carbon 5 side chain of a bile acid, and to put a carboxylic acid on. So you need a 27-hydroxyl to lose propionic acid to generate a C24 bile acid. So in the case of CTX, as we all know, it's due to, I'm missing the A1, but it's due to a deficiency in the activity of CYP27A1, and you don't make it. Now, the actual underlying pathogenesis of the disease was very controversial many years back, but I mean, we all accept that it affects predominantly the chenodeoxycholic acid pathway, and the reason for that is that when you block this 27-hydroxyl, you don't get this, you don't get the thing. What happens is that a 25-hydroxylation pathway kicks in, and followed by 24-hydroxylation to yield cholic acid. So what you find is that cholic acid synthesis is maintained, and I think that's why, in the case of the neonate, if they ride through this transient cholestasis, as the bile acid pool expands and the canalicular transporters become more mature, then they get through over the cholestasis, it resolves, and everything goes on fine. And they have a subnormal but sufficient level of primary bile acids to stimulate bile flow.
Why do they get cholestasis if you have this other way to get the cholic acid?
Well, you just don't produce enough overall of total. The bile acid pool size is still reduced. Yeah, no, this is a normal pathway even in healthy infants. Because what happens is because these patients are not producing sufficient primary bile acids to generate a bile flow. They're producing some cholic acid, but not enough to stimulate bile flow. No, it doesn't raise the levels. Those levels are there, and then they eventually, in a normal infant, what would happen is that this pathway would become less important. And this was originally called the Yamasaki pathway, but it becomes less important because the classical pathway kicks in and produces most of the normal bile acids. So, but that's not going to happen in the case of CTX, because you can't cleave the propionic acid. You don't 27-hydroxylate. So yeah.
You don't have 27.
Because the canalicular transporters are underdeveloped. So they have a stress situation, and then you have suboptimal, a small bile acid pool, and you don't have sufficient bile acid to stimulate bile flow. The major role of a primary bile acid is to stimulate bile flow. And so in bile acid defects, when you don't make primary bile acids, you have no stimulus for production of bile. So the substrates and the intermediates accumulate within the hepatocyte, and that's what's causing the liver damage. Okay, and the liver damage we was discussing this morning, I mean, and I think this points to the real need for having early diagnosis and having this in the neonatal screen, is that it can actually be so severe that it is fatal. And this is a publication that we had a few years ago in a collaboration with Fudan Children's Hospital in Shanghai. And this was 8 infants, you can see the ages there, 2 to 7 months, presenting with severe cholestasis. And they all had mutations in CYP27A1. 4 died of end-stage liver disease at ages 5 to 9 months. 1 underwent a liver transplant at age 11 months, and 3 eventually did clear their jaundice. But the other thing that was interesting was when they delved into the clinical history, in 3 of the families, they reported multiple spontaneous abortions, and I think you brought this up earlier, somebody did, and there were 2 sibling deaths. So I think the thing is that a substantial proportion of CTX patients with severe neonatal cholestasis may well die or need a liver transplant. So that really, for me, points to the real need to get this into the neonatal screening program. So, and in fact, you probably might want to cite this paper, you know, as some of the evidence when you put forward the application. But So the diarrhea, what we say, as I've said, suggested secondary to reduced bile acid pools and the possible cathartic effect of bile acid. And you see here, this is from Jerry Salin's early work, but you can see the pool sizes. Keno is affected much more than colic, and that's for the reason I've just shown you through the pathway. Okay, next slide. And here you see the fecal bile acids in a patient, and you can see a phenomenal number of different bile acids. They're all characterized by structures that lack a 27-hydroxyl. So in terms of treatment strategies, I won't go through this in too much detail because I want to really focus on the colic and keno. But the targets for therapy are actually to suppress endogenous bile acid synthesis, the master enzyme being CYP7A1, cholesterol 7-alpha-hydroxylase. HMG-CoA reductase, of course, is the rate-limiting step for cholesterol synthesis. We want to target bile alcohols. We try to reduce those because we believe those are partially responsible for some of the toxicity that we're seeing and the symptomology. And you see cholesterol. And you can see that keno is very effective at all of these except for either it has no effect or it does downregulate cholesterol synthesis. Colic is similarly effective as keno. So the rationale for colic versus keno is something we, in the pediatric population, we battle with. And what I've done here is try to summarize the pros and cons of using either. I mean, the first thing with colic acid is, first of all, it is approved by the FDA for the treatment of all bile acid synthesis defects, well, not all, sorry, with the exception of amidation defects, but including CTX. It's a highly hydrophilic bile acid, and from that perspective is extremely what we call membrane friendly. It doesn't show toxicity towards membranes. There's been no known hepatotoxicity associated with colic acid, and it's extremely well tolerated in infants and children. The objections to using colic have always been, and I hear this many times, its affinity for FXR is not very good. Well, what I want to show you is that that may be true depending on how you look at the data. Let me say that it's marginally less than for keno. The pros for keno is that it has, as we know, a long-standing track record of use in adult CTX patients. Its affinity for FXR is greater than that of colic in vitro assays. And the cons are, I think, are that it's a hydrophobic bile acid and it has side effects which have included hepatotoxicity and diarrhea, and it's less ideal from that perspective for neonates and infants. And I'm not talking specifically, I'm being general in terms of these properties of keno, not specific to CTX. So, but let me look at this affinity thing, because I just want to make one point about it, is that when you look at the binding affinities of colic and keno for FXR, there's no question that many of the papers, a vast amount of literature, shows that keno is much better has a much greater affinity for FXR than cholate. However, one of the issues with that is that in most of those cell lines, they're using cells that lack a bile acid transporter. And so, however, if you now transfect those cells with a liver-specific bile acid transporter and then retest the affinity of colic and keno, as you can see in the right panel here, then you find that colic and keno stack up very nicely and are very similar in terms of their ability to act through FXR. So, and I'll show you that with some actually clinical data in a moment. Okay, the other supporting evidence that colic is very effective at suppressing bile acid synthesis through FXR is actually from our early studies on other bile acid defects. And this is the most common one, the HSD3B7 deficiency. And you can see this is the data that we submitted to the FDA, the first lot of data from the first 19 patients that were treated with colic acid. And you can see it very effectively normalized bilirubin and transaminases. And actually, I don't have the slide. I've just realized I didn't put it in. But it actually normalizes and reduces the levels of the atypical bile acids. You can see the effect is quite impressive on growth, and it normalizes liver function. I mean, we've done very few liver biopsies because the IRB considered the treatment to be so spectacular that they couldn't justify doing follow-up liver biopsies. So, but that's one of our early follow-ups. You can see here a normalization 16 months after treating with cholic acid. So clearly it is effective. But one of the things that I was asked to talk about combined colic and chemo, but to my knowledge, there have been no studies on combining colic and chemo in CTX, and it's maybe something that might be worth considering. But for the neonate, I think it's not a good idea, because our early experience of these defects is that the initial thought was, when we discovered the first defect, the HSD3B7, we thought, oh, the idea was we basically tricked the liver into believing it's making primary bile acids, and the bile acid pool is almost equal amounts of colic and chemo. So our initial approach was to use a 1-to-1 ratio of colic and chemo, and actually it created some problems with these young infants, these neonates. And a couple of the problems were that it caused a rise in transaminase. Here you see a rise in ALT. Actually, this child was first treated with urso for cholestatic disease, the vitamin U for cholestasis. And you can see here there was the expected decrease in alkaline phos. And then when we made the diagnosis of a bile acid defect, we put the 1-to-1 ratio, and you can see here now the rise in alkaline phos. The other problem was the cathartic action. When we combined colic and chemo, we discovered that these infants actually developed a watery diarrhea that was quite severe and you see here, they've got these lesions on their buttocks. And so what we did, we dropped the chemo, being the more hydrophobic and more hepatotoxic bile acid, and continued with monotherapy. And these issues completely resolved themselves. So the cathartic effects of bile acids have been known for a very long time. They have both propulsive effects they affect motility, and they have secretory properties. And you can see it actually goes back to 1938, one of the first observations. So, and this is also seen clinically, because when you have a situation of increased bile acid loss into the colon, it causes bile acid-induced diarrhea. And cholestyramine, as you can see from this paper, has actually been used to treat constipation, because it increases bowel movements and leads to a more watery stool and in some cases diarrhea. When you look at the secretory activity, dihydroxy bile acids have about twice the secretory activity of trihydroxy bile acids, and chemo is a dihydroxy bile acid and colic is a tri. And there's also the detergenicity, which I've mentioned repeatedly, is much greater for chemo than it is for cholate. So I'm going to skip because I know we're behind on. I'm going to skip a couple of slides and just jump forward to show you just maybe one case situation here that colic, to convince you that colic really does work. I think we know that anyway from your studies from Israel as well, and several other publications that have more recently appeared in the literature. But you know, this was a patient was a boy diagnosed at just over 8 years of age with CTX, and we treated that patient with colic acid. And you can see here, based on the urinary FAB-MS, you see the very high levels of bile alcohol glucuronides that are excreted in the urine, consistent with the diagnosis of CTX. And now we do post-treatment. Did I say post-treatment? Yeah. And post-treatment now, these have essentially disappeared or got down to very low levels. And you can see, but based on a quantitative assay, you can see here, these are the actual concentrations. And to make the point that, you know, that colic acid is also effective at lowering cholesterol levels. This is the cholesterol levels that were in follow-up on treatment with escalating doses of colic acid. So it is effective, and we know that from more recent data from other studies. So while I said I don't do neonatal screening for these patients, but I pick up a lot. This was a slide I showed a year ago. We have a number of maybe 3 or 4 more patients since I made this slide. But so we're about 70 patients, and this shows you the age distribution. And I'm picking those patients up, you know, by our screening method, primarily because they're being filtered out. They're being investigated for idiopathic cholestasis, and you know, they're not thinking CTX. They're thinking could it be a bile acid synthesis defect, of which CTX is a bile acid synthesis defect. But you can see the age distribution of those nearly 70 patients are shown here, and 42% of the cases that I've picked up now we diagnosed in the first 2 years of life, with the youngest one being at 2 days of life. So this is the bile alcohol levels based on our method, which is very similar to the method that the rest of you use. But this is for urine, not for blood. I don't do blood spots for CTX. But and you can see here that the CTX patients have markedly elevated urinary bile alcohol levels. This is just one of the bile alcohols. It's similar for all of them. And then you see the range of levels in 16 patients that we followed that were treated with cholic acid, and 10 patients that were treated with Keno. So the 26 patients there, and I want to just show you the paired samples. So the pairing, this is baseline after compared to post-treatment. You can see here that both cholic and Keno cause a normalization in the bile alcohol levels. This is that's a logarithmic scale. And if you lay them on top of each other, there's not a lot of difference. So both bile acids, cholic and Keno, effectively reduce bile alcohol excretion consistent with reduced hepatic bile acid synthesis. And the only thing I would say is that if you do the calculations here, the average percent reduction in bile alcohols was 70% for cholic and 90%. So marginally greater for Keno. And I think that pretty much fits, with the affinity studies, the binding affinity for FXR. So the other issues for infants, I think, and I stress this for infants as opposed to adults, is the issue of the use of Keno could well be problematic, and was problematic in the other defects that we've treated. And there are documented. There was from old data, the National Cooperative Gallstone Dissolution Studies. They documented increases in serum transaminases even in adult patients with gallstones that were treated with Keno. Now, keep in mind, they're not like CTX patients who don't make Keno. These patients make Keno and make cholic. So what's happening is they're piling on more Keno. So now you get to really high levels of Keno. So it's maybe not surprising that some of those patients present with elevated transaminases. But I show you the other two paper here. This one you're familiar with by Heidi, that this Keno is highly hydrophobic and intrinsically hepatotoxic. So, but we also showed this is the case for the other bile acid defects, particularly the 3-keto, the AKR1D1, the delta-4 3-oxosteroid 5-beta reductase. What we've treated, though, when we treated those patients with Keno, and we don't treat them in the US with Keno, we treat them with cholic, but this is from our China experiences in China, Fudan Children's Hospital. Keno was effective, but only at low doses. And in fact, when we dose them at what would be the recommended dose of 10 to 15 milligrams per kilogram, then it was actually toxic to those. So so what we do in those patients, if you can't use cholic and have to use Keno, it's important to titrate to make sure that you actually don't get this toxicity. So I think probably in the interest of time, I'm going to skip and say in terms of cholic acid, the safety, it's it has been singled out as curative for bile acid synthesis defects. And we've seen no drug-related adverse effects in more than 25 years of use, and actually nearly 30 years of use. It gives impressive clinical responses in bile acid synthesis defects, normalizes liver biochemistries, resolves histologic abnormalities in those patients we've been able to do follow-up liver biopsies, and improves growth. And we've had very few treatment failures as a result of cholic acid. So I think at the end of the day, for me, my belief is that, I don't know if I got one more. No, I don't have one more slide. I think, let me go back. Oh, can't go back. Yeah, I think that it has particular advantages for use in the neonatal presentation of CTX. And I think I'll end on that. Okay, I'm happy to take some questions if you have any.
Well, thanks very much.
Okay.
And you know, again, I think it's great to have different options. Well,
Yeah.
I think in that the Dutch paper, they lowered the dose to about 5 milligrams, and that alleviated the hepatotoxicity.
Yeah, I mean, most of these were between 4 and 6 milligrams. So you say 5 milligrams, which is much lower than what we use for colic acid.
So I do have one question. So you've been identifying these, infant cholestasis cases, CTX, for how many years now?
Oh, gosh. When did we start the program? 1988? Since 1988.
Presumably they went on treatment, and you showed one follow-up of 5 years for one of your cases. So for the newborn screening, one of the really important things to try and show favourable outcome for long-term treatment for children identified very young. So I know you're busy with many.
I mean, some of these patients have been on much longer than 5 years. In fact, one of the other examples I had was, I think, 14 years.
So would you?
I can't remember the longest because I'd have to go back and dig into the notes.
So we need to go back and maybe try and contact some of the physicians or the patients and see if you can
Yeah,
get more data. Because we don't have much data to publish, you know.
Yeah, no, I'm certainly willing to. It's a question of the resources to do it. I mean, I can tell you from experience when we compiled the NDA for colic acid to the FDA, trying to track down these patients years later when they're scattered all over the place. If this was a common disease and they were in our institute, it would be a lot easier. But you know, it isn't easy because you've got to get hold of the primary physician, and often the primary physician's moved on to somewhere else and the patient's relocated. And I mean, for example, a couple of patients we've had where their treatment response was spectacular, and then they disappeared, and then they re-emerged with a massive liver and spleen, and they hadn't been taking therapy and in another hospital, and I re-diagnosed them. So this is a sort of problem in trying to track down over that length of time. But yeah, if we have the resource, I think we could try to do it. Yeah. Yeah, I mean,
sorry, the efficacy of treatment should be monitored over time. So patients should be having a test, ever so often to just make sure that their biochemistry is normal. So how many of those, like,
yeah, well,
repeat
I mean,
samples
that
do
is
you
the
get?
26 that I showed you. I mean, out of 60, nearly 70 now, I only have the follow-up on 26 of them.
So you have follow-up on 26
Yeah.
though,
Yeah.
that's pretty good.
Yeah, that's okay. You know,
Any other questions?
it's like money, you know, the more you have, the better. You know? So you can get more patients, although you don't really want to get more parents. You'd rather patients not have this disease. So, but yeah, you know, it is extremely difficult doing follow-ups, you know.
So are there other organs that for which CDCA or colic acid are toxic? You just mentioned the spleen. What about the kidney? Are any other organs measurably affected by additional too much CDCA or colic acid?
Well, I can't think of any specific examples of publications of that, but I mean, you know, if you're working in cell culture and stuff like that, people use bile acids because they are detergents and they use them to break down tissue. And the more hydrophobic the bile acid is, the easier it is to break down the tissue. And so, you know, in the case of other organs, I mean, the liver is the primary site of bile acid synthesis, and that's where, you know, you would expect that to be. But the kidneys are filtering bile acids, and I wouldn't like to say that they're not toxic to the kidney, but that's not an area that I'm aware of has been studied in any detail. Bob, do you have any experience of that kidney? Yeah.
Okay, well, thanks very much.
Okay, thanks.
There's also a question in the chat. I was hoping that maybe Jean could read that
Sorry,
and
there was
ask the
another
question. Yes.
Oh, there's a Zoom question.
It's in the chat.
Yeah, I'm sorry. I didn't see the Zoom question. Since taking CDCA suppresses colic acid, would it be helpful for symptoms control to take both?
As an adult?
So yes, it is an adult.
Possibly. But, you know, I say that based on just speculation. I mean, no data. There hasn't been a systematic study done that I'm aware of, using both a combination of colic and keno in CTX.
But you have used it for the baby that you
Yeah,
showed
for the
with
babies, and
with
we
the
stopped
severe-
using it because of the reasons I showed you. Yeah.
So
We also tried urso, a combination of urso, because it's a potent choloretic, thinking that, it is like we joke in hepatology, it's the vitamin U of hepatologists, and thinking because of the potent choloretic action, and it does effectively lower transaminases, that if we combined urso and cholate, but actually what happened was that when you do that, urso interferes with the absorption of cholate. So we dropped urso. Cholate works well on its own. But I think in CTX, maybe the thing to do is to use a combination. The early work in the 60s showed that colic inhibits keno synthesis and keno inhibits colic synthesis. It was work from Henry Danielson's group back in Stockholm in the late 60s. So.
but
Just
to
as a
this point,
comment,
nothing's
there
been
are
published on that, but
yeah, but
maybe
there's no, I mean, one would have to design a study. I mean,
yeah,
there
whether you take, maybe what you could do is you take a patient already on keno and you then continue to follow them, but you throw in cholate. I don't know. Maybe you should start with, new patients, newly diagnosed patients. But I mean, it's difficult to assess, actually.
yeah, because
are two of us here who have started on colic
absolutely.
after doing CDCA.
publications needed on that clinical trial. So it's my pleasure.
So I don't have a good answer for the question, because it's hard to know when you have no data. But
Thanks very much, Ken.
okay, there you go.
My pleasure to introduce Dr. Austin Larson, who will be talking about the results of the RESTORE clinical trial.
So thank you all for having me. I'm presenting on behalf of all of the site investigators for the RESTORE trial. This is a trial that was initially supported or sponsored by Travere, and the trial was taken over by Mirum. Many of the individuals here are employees of either Travere or Mirum, and all of the investigators had financial support to conduct the trial. So by this point in the afternoon, I don't need to tell you too much about the background of CTX, other than to say that it's an important clinical problem to address. A lot of folks in the room here are either themselves or in a family member dealing with the symptoms of CTX, and we're all really motivated to identify effective treatments and to establish exactly which symptoms those are effective for and over what time period. I also don't need to give you much background on bile acid synthesis at this point, other than to just bring you back to the idea that CTX is a deficiency of an enzyme that prevents the formation of the mature bile acids. As a result, there's both a deficiency of those bile acids that's probably responsible for some of the symptoms, and then probably the primary mechanism is the accumulation of toxic intermediates that keep getting pushed into this pathway due to the lack of negative feedback from mature bile acids. Patients with CTX traditionally have been diagnosed with elevated cholestanol, lack of mature bile acid production, production of atypical intermediates that don't accumulate in the absence of the enzymatic block. And the goal of therapy traditionally has been to normalize cholestanol by establishing that negative feedback from the mature bile acids. Standard of care being chenodeoxycholic acid, but as we also learned, cholic acid also has that negative feedback effect on the pathway. Some case histories that will probably resonate with some folks in the room here. These are taken from Dr. Dwell's case series published in 2018. These images show you some of the xanthomas that can appear on the Achilles tendon or on the extensor surfaces of the knuckles. These are both patients that were treated with chenodeoxycholic acid and had stabilization of their neurologic symptoms after treatment. But all of the evidence that we have is in a non-randomized form. So the medication CDCA became standard of care without a placebo-controlled trial. Dr. Eichler is not in the room anymore, but he gave us the introduction to clinical trials that ideally we would have placebo-controlled trials at a time when we have equipoise, right? That we don't have a clear understanding of some benefit of a treatment. Once we've established through non-randomized types of trials that there may be benefit for a therapy, it's difficult to ethically design a clinical trial that will establish the superiority of a medication over a placebo. So cholestanol is a biomarker, and as we'll hear in the next talk, it's also mechanistically related to the symptoms that folks have with CTX. So cholestanol is both measurably elevated, but we also know that it is part of the damage that occurs to tissue. We can find it in those xanthomas. We can find it in brain tissue. And we know that it is part of the pathogenesis of the disease. So this is a reasonable thing to look at as an outcome for a trial, recognizing that the bar is pretty high for a biomarker to be the endpoint for a trial. Chenodeoxycholic acid, as we've been hearing about from many folks today, it is an FDA-approved medication. It was approved in 1983 for the treatment of gallstones. It has been recognized for decades now as the standard of care for CTX. And with non-placebo-controlled trials, the understanding in the community is that there is benefit of using that treatment. However, CDCA is not FDA approved for the treatment of CTX. So the prior manufacturer of CDCA had a discussion with the FDA about what the structure of a trial would be, given that we're in this circumstance where CDCA is understood to be the standard of care, and yet it got to that position without a placebo-controlled trial. So that brings me to the design of the RESTORE trial. So the RESTORE trial was designed with the understanding that CDCA is standard of care, that most patients would be on CDCA at the time of recruitment into the trial. So this is a bit of an unusual design for a drug trial, which is that patients are identified by clinicians, by investigators on the trial. They're screened for eligibility for the trial. And then all patients undergo 8 weeks of open label treatment with CDCA. Then half the patients are randomized to placebo. Half the patients are randomized to CDCA for a 4-week period. They're monitored closely, monitored weekly over that period of time to establish the biochemical and clinical differences between patients on CDCA and those patients on placebo, both in terms of changes in biomarkers that we know are part of the pathogenesis of CTX, but also safety monitoring, things like liver function tests, which we heard about in the last talk. Then all the patients go back onto open label CDCA. And then there's a second double-blind period where all of the patients cross over. So all of the subjects in the trial have a 4-week period on CDCA and a 4-week period on placebo. but the investigators and the patients are blinded to whether they're on placebo or drug during the crossover period. 30 days after completing double-blind period 2, there's a phone call check-in for the clinical status of the patient. The dose of CDCA was 250 milligrams 3 times daily for all patients. And right now I'm telling you about the adult portion of the trial. So this is 14 individuals. There were also 5 pediatric patients, but we're not presenting those data today. The trial was designed with a rescue option during the double-blind periods, and the rescue to active drug could be triggered either by clinical concerns of the investigator that a patient was clinically worsening while on placebo, or there were biochemical criteria that if a patient had a significant enough elevation in CTX associated metabolites, they would then revert back to active drug. So the primary endpoint for the trial is bile alcohol, specifically the change in urine 23S pentol. Secondary endpoints included some of the metabolites you've heard about today, including 7-alpha C4, which is also referred to as C4, 7-alpha 12-alpha C4, cholestanol. So those are the abnormal and potentially toxic intermediates that are accumulating in CTX patients. Another secondary endpoint is the proportion of patients on either the placebo or the drug that for whom the rescue was triggered during those double-blind withdrawal periods. And then safety endpoints were monitored, including, and most importantly, liver function tests. Another exploratory endpoint was to look at those bile alcohols, both in plasma in addition to in urine. As I mentioned, this is the adult cohort of the trial. These are individuals that are 16 years or older, pertinent to Dr. Felix Sakai's discussion. So these are women who agreed to use reliable birth control. And then these are all symptomatic patients. So these are individuals that have at least one symptom of CTX. They had a biochemical diagnosis, and as it happened, they all had a genetic diagnosis as well. So this is a diagram showing you where these measured metabolites are in the pathway. Again, CDCA is the treatment drug. It decreases the flux of cholesterol into the bile acid synthesis pathway. If that inhibition is relaxed or removed by the patient going on placebo, then you would expect a fairly rapid increase in the metabolites that have gray boxes there. And that is indeed what the trial showed. So in almost any paper, one of the big questions is, was the population that enrolled in the trial, were they representative of the population at large? And the answer in this case is yes, the population was fairly representative of the American population overall. I should mention some of these patients were also in Brazil. They are 9 males and 5 females. The racial and ethnic background of these individuals is roughly representative of the country at large. And again, all of these patients had a confirmed diagnostic genetic result showing the genetic diagnosis of CTX in addition to the biochemical diagnosis. As we've heard about in a lot of studies, the mean or median age of diagnosis was 35. So that seems to be something that comes up as kind of the typical age of diagnosis. The disease burden for these patients was also fairly similar to what we've seen in a lot of CTX cohort studies. So 78% of the individuals had learning disability, 71% with ataxia, 71% had also experienced some cognitive decline at some point in their lives. 64% had brisk reflexes. That same number had xanthomas. So I think when you look at this, you'll recognize that this is, again, fairly representative of the population of patients that we care for with CTX. One of the things that was interesting to me, the patient that I cared for in this trial had kidney stones. And looking at the literature, right, there are 800 patients with CTX published. There are only 2 case reports that I could find of individuals with kidney stones, but there were 4 folks in this trial with kidney stones. So I'm interested in that as a potential signal, something to follow up on here. So getting to the results. So this is the primary endpoint. This is the urine 23S pental level. And the main finding to draw your attention to, so there's 2 cohorts here. The red cohort had withdrawal of drug and started placebo at day 1 after the run-in period. And you can see for those individuals that were on placebo, there was a rapid rise in 23S pental, and that is quite a statistically significant difference, even starting early in the withdrawal period. And then if you look at the second cohort that had withdrawal of drug during the second period, you can see the exact same pattern. And the folks that stayed on active CDCA are the mirror image of that pattern. So this is an indicator of a rapid 20-fold increase in the 23S pental, that abnormal bile acid. Looking at cholestanol, there is a statistically significant difference between the 2 groups, but you can see that the difference is much less of a marked difference. This is a 2.8-fold increase during the placebo period as opposed to the active drug period. And it's certainly not as differentiated as the 23S pental. Again, that is statistically significant. And it's important because cholestanol, we've established, is likely part of the pathogenesis of CTX. And it is the metabolite that has by far the longest track record in terms of being associated with CTX. Moving on to some of the other metabolites, an even more substantial difference was seen for 7-alpha-C4 upon withdrawal of the active medication. So in this case, that was a 50-fold increase, and it started very early during the withdrawal period. And a similar pattern was also seen for the 7-alpha-12-alpha-C4. Lastly, the plasma bile alcohols also have the same pattern. And again, for each of these, it's to a greater or lesser degree in terms of the difference between the placebo group and the drug group, but it is clearly the same pattern for each of these metabolites. One of the other secondary outcome measures was the number of patients that required a rescue during their placebo, either their placebo-controlled period or their active drug period. So 2 out of the 7 patients during the first period of placebo were rescued to the active drug during that 4-week period. During the second period, it was all 6 of the patients that were randomized to placebo, as well as 1 of the 7 patients that was on active drug. Now, of those rescues, only 1 of them was a clinical rescue. So it was a clinician that felt like they identified worsening ataxia in a patient and rescued them to active drug. The 1 patient that was on active drug and underwent rescue, that was an intentional overdose by the patient. So this is kind of a technicality in that the patient then entered the open label period earlier, but they didn't actually have that's not a clinical worsening during the active drug period. In terms of adverse effects of the drug, all of them were judged to be mild or moderate. They included diarrhea, constipation, and headache. There was 2 of the patients who had constipation had it while receiving CDCA, and 4 had that at any point during the study. The other side effects were seen throughout the study. So the conclusions from this study, this is the first randomized double-blind placebo-controlled clinical trial evaluating CDCA withdrawal. The structure of the trial was due to the establishment of CDCA as standard of care for many decades. And the findings of the study were that the withdrawal of CDCA treatment rapidly resulted in statistically significant increase in multiple different biomarkers, urine 23S pentol, plasma 7-alpha-C4, 7-alpha-12-alpha-C4, cholestanol, as well as plasma bile alcohols. And then conversely, there was also for the folks that were on placebo during the first double-blind period, there was rapid decreases in all of those metabolites when they went back into the open label period of active drug. A statistically significant portion of patients receiving placebo required blinded rescue medication, although the vast majority of those were due to pre-specified biochemical criteria and not clinical worsening. Again, that wouldn't be expected during such a short time period of withdrawal. So the overall conclusion is that CDCA treatment was well tolerated, with most adverse events being self-limited and mild to moderate, and there were no abnormal liver function tests deemed to be clinically significant during the trial.
That was a great talk. Thanks very much. Any questions?
The patients appeared to do worse in the second double-blind period compared to the first. Is there any logical explanation for that?
So you're talking in terms of the rescue or in terms of the magnitude of change in the metabolites?
So 6, if I'm understanding, if I remember, 6 out of 6 in the period had to be rescued. Those were all, or almost all, biochemical rescues. So yeah, I'm interested in the rescue.
Yeah, so I don't know the answer to that. I may speculate a little bit. Yes. Okay, great. Before I speculate, let me let you say something. Well, I was going to say, so some of the patients were treatment naive heading into the initial open label period. So you can see, like, for example, in the cholestanol period, some of these patients were treatment naive, and it was sort of taking them time to chemical control of their disease from many years of not being treated. So that would be my speculation, but I'm curious of what you think.
So the rescue was on the 23S pentol biomarker. So if you go back to that one. So as Austin said, some of the patients were treatment naive and coming into this. So I think it's just a reflection of the ones in the second double-blind period had a longer time to reach the lowest baseline. So they were looking at a tenfold increase in the baseline. So at that point, we were maybe able to discriminate that a little bit better than at the beginning when it was still coming down to the baseline. But that's just a postulation.
Yep, I think we're saying the same thing there. Yep.
Osteoporosis or osteopenia on the list of. Can you put the microphone so that people on the can hear you?
I'm wondering about osteoporosis and osteopenia in these 14 individuals. It wasn't on the list there. And then second question, why does CTX Why can it be part of the cause of those conditions?
This is the most commonly reported medical history for individuals with CTX. These are individuals that are at an average age of 39. So many of the phenotypes or many of the clinical manifestations of CTX are things that are age dependent. So they happen earlier than someone without CTX, but they still don't happen early in life. So, you know, I think it's just, you know, that's not something that had been captured by this point in folks' lives. Yeah.
Yeah, because one of the features of biological synthesis defect, sorry, yeah. I keep forgetting on Zoom, but yeah, the question was, did you measure vitamin D levels? Because one of the features of bile acid synthesis defects, not in 35-year-olds necessarily, but in the neonate, is that because you don't produce a high enough concentration of bile acids in the intestine, then you have fat-soluble vitamin malabsorption. In fact, in the amidation defect, that's the major phenotype, rickets and fractures. So I don't know, in this case, that could be a possibility for osteoporosis if it's a reflection of fat-soluble vitamin adsorption or status. So I don't know. Did you do that or not? Probably not.
I don't believe so. I can't say for certain, but I don't recall that. Yeah.
For adults?
So the question was whether the study was only for adults. So there was also a pediatric cohort. So there were 5 children that were enrolled. That data has not been analyzed yet. So it took longer to enroll the 5 pediatric patients. They're not included. This is only the adult cohort.
But the kids weren't taken off medication.
Correct. Yeah, there
So
was a
slightly different.
There was a different design for the pediatric trial. So it hasn't been analyzed yet. Yeah.
Okay, well, great. Well, thanks very much again. Yeah. Okay, we're in the home stretch. So I'm excited to announce the next speaker, Dr. Zhang, who's joining us from China. And so we're kind of, we finished all the talks now about clinical trials, and we're kind of changing tack a little bit for this last final talk. And so really not much work has been published in the literature. We think that there's toxic effects of the cholestanol and the bile alcohols, but on the mechanism of how this causes toxicity in the body in CTX. So Dr. Zhang published very recently some very basic science research work that's very interesting, looking at the mechanisms of toxicity of cholesterol. So welcome, Dr. Zhang. Thanks for joining us.
Hello, it's really my great honor to be invited to this wonderful meeting. Unfortunately, I cannot go to America to attend this meeting, so I have to do this online. Can you hear me?
Yep, we can hear you and we see your presentation.
Okay, very good. So as you just mentioned before, here I'm going to talk about basic research regarding how the CTX patient have Parkinsonism. So the title is cholestanol accelerates alpha-synuclein aggregation by activating aspartate peptides. My name is Zhentao Zhang. I work in the Department of Neurology in Renmin Hospital of Wuhan University. That's in central part of China. So I think the background of CTX has been talked very much today, so I won't pay a lot of time in talking about this background. But cholestanol, the levels of serum cholestanol is increased in those patients. And those patients usually have neurological or neuropsychiatric abnormalities. Especially Parkinsonism. Parkinson's disease is another disease. It is one of the most common neurodegenerative disease. It usually happens in the elderly, and the main pathology is the loss of dopaminergic neurons in the substance nigra and the formation of Lewy bodies in the remaining neurons. Unfortunately, the etiology and the pathogenesis of Parkinson's disease are not fully understood, but aggregation of alpha-synuclein plays a key role in the pathogenesis of Parkinson's disease. Since the alpha-synuclein is expressed mainly in the neurons. However, in the brain of Parkinson's disease patients, it forms oligomers and then fibrils. And the fibrils can act as seeds to trigger the aggregation of more alpha-synuclein and making more and more fibrils, and finally destroy the dopaminergic neurons in the substance nigra and causing motor impairments such as static tremor, bradykinesia, and abnormal gait. So Parkinson's disease is characterized by this aggregation of alpha-synuclein, and a lot of CTX patients will have Parkinsonism. So we ask whether there is some relationship between CTX and Parkinson's disease. Actually, it has been reported that the CTX patients show dopaminergic pathway degeneration and Parkinsonism. Cholesterol deposits in the substance nigra in patients with CTX, which is also affected by Parkinson's disease. So we think there may be some relationship between them. Previously, we reported that an enzyme named aspartame endopeptidase, AEP, the gene name is LGMA. It cleaves alpha-synuclein to form an alpha-synuclein fragment, alpha-synuclein N103, and promotes the aggregation of alpha-synuclein and Parkinsonism. So also the expression of this enzyme AEP is regulated by the transcription factor CAP beta. So we reported that this transcription factor CAP beta binds to the DNA and promotes the expression of AEP, and AEP cuts alpha-synuclein, causes the fragmentation of alpha-synuclein, and promotes its aggregation. So we asked whether the CTX patient, in the brain of a CTX patient, this pathway, this CAP beta AEP alpha-synuclein pathway is also activated. So about 5 years ago, we had a patient came to us. She was 47 years old at that time. The chief complaint was bradykinesia and static tremor for 4 years. She was previously diagnosed with CTX. He already taking CDCA, and his Achilles tendon mass has been removed. A physical examination showed that this patient has Parkinsonism. So we performed a gene examination and found this patient had compound heterozygous mutation of the CYP27A1 gene. We also examined the gene of her parents and found one of them came from her mother, and the other one was from her father. And we also performed PET scanning of the tendon and found that there's a enhanced metabolic activity in the Achilles tendon, and also a pathological examination showed that there's a accumulation of cholesterol-like substance in the Achilles tendon. We also tested the concentration of cholesterol in the serum and found that the concentration is highly increased compared to control. So I mentioned, I mean this lower peak, this CTX patient is much higher than the control. And also we performed MRI scanning of the patient and of different time. We can see that there's abnormalities in the cerebellum and the middle brain, and also the also in the cerebrum. So since this patient has multiple intracranial lesions and also increased cholesterol, so thus we performed PET scanning of the DAT PET scanning. The DAT means dopamine transporter. It is a marker of dopaminergic neurons. Usually in the patients, in Parkinson's disease patients, the levels of DAT is decreased. So we also found in this patient, we also can, the PET scanning showed decreased DAT signaling, means the dopaminergic neurons is degenerated in the patient. We also see, we also performed neuromelanin MRI scanning and found decreased neuromelanin content. All these scannings shows degeneration of dopaminergic neurons in the substantial striatal pathway. So indicating this patient have something very similar to the sporadic Parkinson's disease. So thus we asked whether the concentration of cholesterol is also increased in patients with Parkinson's disease, with sporadic Parkinson's disease. Although this patient do not have a CTX, but we can see the levels of cholesterol is higher in sporadic Parkinson's disease. So does this cholesterol cause neurotoxicity? So we cultured SHSY5Y cells, and we added different concentrations of cholesterol, and we tested the cell viability. Interestingly, we found that cholesterol reduced the cell viability of cultured SHSY5Y cells, indicating cholesterol is toxic to cultured cells in a concentration-dependent manner. So next question, we asked whether cholesterol can activate the AEP, which play a role in the pathogenesis of sporadic Parkinson's disease. And thus we cultured SHSY5Y cells, and we treated the cells with cholesterol, different concentration, and we tested the expression of the CB beta, this transcription factor, the AEP, this enzyme, and also the product of AEP, this N103 alpha-synuclein fragment. We found that all these transcription factor, enzyme, and the fragment all increased in a concentration dependent manner after treatment with cholesterol. So indicating cholesterol can activate this CB beta AEP pathway. Since previously we reported that this CB beta AEP signaling pathway promotes the aggregation of alpha-synuclein. So we also tested whether cholesterol can promote the aggregation of alpha-synuclein in a cellular model. The cellular model, this is HEC293 cells. These cells express GFP tagged alpha-synuclein. Once we add alpha-synuclein fibers, this is alpha-synuclein preformed fibers. Once we add the fibers, alpha-synuclein forms aggregates. But in the presence of cholesterol, alpha-synuclein forms much more aggregates compared to the group without a cholesterol, indicating cholesterol not only activates the CB beta AEP pathway, but also promotes the aggregation of alpha-synuclein. So these red dots are phosphorylated alpha-synuclein, presenting pathologic alpha-synuclein, and we also see the phosphorylation of alpha-synuclein is also increased. So we also showed that this alpha-synuclein can promote the alpha-synuclein aggregation is increased by clostanol. So we asked whether AEP, this enzyme, participate in this process. So we used this shRNA methods to knock down AEP to show whether this can block the toxic effect of clostanol. Yes, we did. We found that this active AEP is decreased by knockdown of AEP. We also showed that this AEP inhibitor, compound 11, this is a compound that we found to inhibit alpha-synuclein, to inhibit the activity of AEP and alpha-synuclein aggregation. We found that this compound 11, AEP inhibitor or knockdown of AEP can attenuate the toxic effect of clostanol. Also, we showed that knockdown of AEP can inhibit the aggregation of alpha-synuclein. Also, this compound 11, this compound can inhibit the fragmentation of alpha-synuclein and phosphorylation of alpha-synuclein, and also this aggregation of alpha-synuclein. So thus we asked whether we further asked whether in animal models this CB-beta AEP pathway also participate in clostanol-induced neurotoxicity. We had this mouse model that once we inject these alpha-synuclein fibers into the stratum, it will seize the endogenous alpha-synuclein and propagate throughout the brain. So this is a well-established model published in about 10 years ago. So we just injected this mouse. We just injected the water mice with alpha-synuclein fibers, and we also the mouse were treated with clostanol, or some of the mice were AEP knockout. So we had PBS group and the alpha-synuclein fiber group. We also have the wild-type mice and AEP knockout mice. Some of the mice were treated with vehicle, and some of them were treated with clostanol. So we have 8 groups of mice. We want to see whether clostanol can promote alpha-synuclein aggregation and neurotoxicity. Also, want to ask whether knockout of AEP can attenuate the toxic effect of clostanol. So briefly, we found that in the wild-type mice treated with clostanol and alpha-synuclein fibers, this is a Parkinson's disease mouse model. We can see the degeneration of we can see this motor impairments. And once these mice were fed clostanol, this the motor impairments was more severe. But in the AEP knockout mice, the toxicity of clostanol was much attenuated by a knockout of AEP, indicating that AEP do participate in the neurotoxicity of clostanol. So next, we also tested the aggregation of alpha-synuclein. We can see that in the alpha-synuclein aggregation mouse model, there's some alpha-synuclein aggregates throughout the brain. But in the mice treated with clostanol, we can see much more alpha-synuclein aggregation. But in those with AEP knockout, we can see this aggregation are much decreased, indicating clostanol promotes alpha-synuclein aggregation, which was blocked by the knockout of AEP. So we also performed immunochemistry to test the innervation of dopaminergic neurons in the stratum. We can see that in patients with that the mice fed with clostanol, we can see much weaker signal, and in the alpha-synuclein knockout mice, in the AEP knockout mice, the degeneration of dopaminergic neurons was much more attenuated. Similar observations was found in in the immunohistochemistry of the substance nigra. The cell body is in the substance nigra, and this cell innervation is in the stratum. In the substance nigra, we can see cholesteranol promotes the degeneration of the dopaminergic neurons, which was attenuated by the knockout of AEP. This is nasal staining, which shows similar results that cholesteranol promotes the aggregation of promotes the degeneration of the dopaminergic neurons, and a knockout of AEP can attenuates the degeneration of the dopaminergic neurons. We also so next we asked whether this since we found that AEP knockout of AEP is protective, but we can knock the AEP in human. So we asked whether this compound AEP inhibitor can also blocks the toxic effect of cholesteranol. So we divided the mice into 4 groups: the vehicle group, the company 11 group, and also the alpha-synuclein PFF plus vehicle group, or alpha-synuclein PFF plus company 11 group. We found that injection of alpha-synuclein PFF triggered motor impairments, and treatment with company 11, this AEP inhibitor, can partially block the toxic effect of alpha-synuclein fibrils. Also, in the other motor impairments, company 11 all showed some protective effects against the toxic effect of alpha-synuclein aggregates and alpha-synuclein fibrils and cholesteranol. So this is the results of alpha-synuclein aggregation. We can see injection of this PFF triggers alpha-synuclein aggregates, and this company 11 can partially attenuate the aggregation of alpha-synuclein. And this is results of both dopaminergic neuron cell body and the deposition of alpha-synuclein. And we also see company 11 can partially inhibit the degeneration of dopaminergic neurons and the aggregation of of alpha-synuclein. And similarly, we performed immunohistochemistry to test the content of dopaminergic terminals in the substance nigra and in the stratum and substance nigra. Both results showed that this company 11 can partially attenuate the degeneration of the dopaminergic neurons and neuronal terminals. This is nasal staining showed similar results. Also we can say that this company 11 partially attenuates the fragmentation of alpha-synuclein, and also the aggregation of alpha-synuclein and this degeneration of dopaminergic neurons. This TH and DAT are markers of dopaminergic neurons. We can see company 11 can attenuate the degeneration of dopaminergic neurons caused by both alpha-synuclein fibrils and cholesteranol. So here's the conclusion. We found that increased cholesteranol may promote Parkinsonism or activating this CAV beta AEP pathway. So AEP may be a therapeutic target for cholesteranol-induced Parkinsonism. And also, more interestingly, we found that even in patients without CTX, in this sporadic Parkinson's disease, the levels of cholesteranol are increased than those with control subjects. Also the molecular mechanisms may be the same, may be similar. Cholesteranol can activate AEP, and as an enzyme, AEP cuts alpha-synuclein, promoting alpha-synuclein aggregation and neurodegeneration. We also developed an inhibitor of AEP that can attenuate the toxic effect of cholesteranol. So this is my group in Wuhan University. We have about 20 members in my lab, and we are working majorly. We're working on Parkinson's disease and Alzheimer's disease, this neurodegenerative disease. And we want to thank the CTX patients and the grants from the National Science Foundation of China. So this is all my presentation. And thank you again for inviting me. And is there any question there? Yeah, thank you.
So thank you very much, Dr. San. That was a very interesting talk. So do you have plans to, so the inhibitor is called CP11, is that correct?
Yes.
Do you have plans to try and do a clinical trial in Parkinson's disease or?
That's a really good question. We want to do that, but company 11 is just a prodrug. This is not a really the pharmacokinetics and pharmacokinetics is not so good. So we are still modifying this compound to make it better, and we are still working on that. Maybe I think this maybe take several years to make this compound ready for clinical trials. I think yes, we are doing that, but it takes a very, very long time. Yeah, thank you.
Well, it's fascinating, and of course we all know that Parkinson's disease can be a symptom or be seen in CTX as well, and we know that cholesterol is elevated in CTX. So this could be helpful, you know, to prevent that symptom in the future if this progresses. I should have one more question. So do you have a mechanism why cholesterol is elevated in Parkinson's disease, sporadic Parkinson's disease? Why would that be?
I'm not quite sure, but I think it's possible that it will affect the activity of this enzyme CYP27A1. We did not test it, but the hypothesis is that cholesterol may affect the activity of CYP27A1.
Okay, but you ruled out CTX in those sporadic Parkinson's disease cases. There were no CTX cases in there.
Yes. Yes, we observe that the concentration of cholesterol are increased, but I'm not sure. I'm not quite sure why this is increased. So sorry,
I'm putting you on the spot.
The mechanism.
That's interesting. Any other questions? I have one back here.
My question is, your Parkinson's patients, are they on levodopa?
Yes, most of the patients are L-DOPA. Yes, I know maybe it is possible that this L-DOPA or other anti-Parkinsonism disease drugs can affect the serum cholesterol concentration. Yeah, that's a very good question. Yeah, maybe we will enroll more L-DOPA naive patients to further confirm this observation.
Any other questions?
Thank you very much for an interesting talk. I wanted to ask whether those Parkinson's disease patients, the sporadic patients, where you found the high levels of cholesterol, did you sequence the gene, the CYP27A1?
Yeah, that's a really good question. Because unfortunately, we do not have a lot of patient samples, but yesterday I was presenting these slides to an expert on genetics of Parkinson's disease, and in China, Dr. Tang, and Dr. Tang also proposed that he will work on this, whether there's any genetic variations in Parkinson's disease patients. Yeah, I think he is going to work on that. Hopefully, we can see some results within this year.
Well, thanks very much again. We'll be watching the research out of your lab and hopefully seeing some of this research progress over the next few years. Thank you very much again. Thanks.
Thank you.
Okay, I think that's it. I'm not sure what time dinner is. Okay, and I'm going to hand it over to Jean.
Well, I'm just happy to announce that Mirum Pharmaceutical has submitted a new drug application to the FDA for Chenodal to be treated for CTX. So it was just submitted last night, late last night, and the announcement came out this morning. We have a group of people here from Mirum, and thank you. And hopefully we'll hear something in about 6 months.
Right. And I look forward to continuing conversations this evening. Yeah, thanks everyone for coming. I thought that was a great conversation.